postdoctoral dissertation from the International Center for Genetic Engineering and Biotechnology at Delhi, which was focused on identifying, just a minute, which was focused on identifying the escape mechanisms of pathogenic mycobacterial strains which have co-evolved with human population. She gained experience in biomarker discovery for susceptible tuberculosis along with next generation sequencing techniques as a postdoctoral scientist at National Institute of Biomedical Genomics at West Bengal. She furthered her research as a postdoctoral scientist at Max Planck Institute for Infection Biology, Germany. Her main focus was on the immune response in various tuberculosis, particularly the neutrophil response and myeloid-derived suppressor cells, which are of importance in cancer immunotherapies. At the institute, she familiarized with preclinical vaccine research methods and worked in biosafety level 2 and 3 laboratories. Her team at BioNTech is involved in the development of mRNA-based prophylactic vaccines against various infectious diseases. And BioNTech has also developed the mRNA vaccines for COVID-19. So dear delegates, please join me in welcoming Dr. Neha Agrawal to deliver her talk on mRNA vaccines. I now request you, ma'am, to lead the session and enlighten us with your knowledge on mRNA vaccines.
Neha, your voice is not audible. Nope. Not audible. Nope.
Ma'am, could you please try to reconnect your audio?
Unmute. Unmute. Yeah, speak, Neha. Speak up. Speak loud.
Now, can you hear me?
Yes, perfect.
Yeah, okay. Yeah, sometimes it happens with different system. So I was saying thanks a lot, Sumati, for so elaborated introduction. I will start with this. Yeah, I'm from Banaras, as you said, and my bachelor's and master's were in Banaras Hindu University. And after that, I left Varanasi. Then I will share the slides.
Yes, we can see that. Mm-hmm.
Can you also see it?
Yes, maybe you can just put it in. Yes, perfect.
Oh, so I changed my idea. Yeah. Okay, so I have a little bit changed the topic called vaccines and mRNA because I realize it is also better to talk about how vaccines work. That means before that, it is good to give introduction of immunology, which is my expertise, and which has enormous opportunities and in scope to improve and learn about as well. So technically, I will talk about pathogens, immunity, stages of vaccine development, types of vaccines, and then finally mRNA vaccine. And just, yeah, I will try to be quick so that we have a question answer session left. So most of the things you might know or not, but it is better to go through and give a little idea. So what kind of pathogens exist in nature? And they have been long before then we arrived on Earth. So if you say like malaria, this is a protozoa, some of the a lot of bacterial diseases is a problem, and these all are prokaryotes. And once we talk about viruses, they are actually acellular, non-living organisms. Why? Because they cannot multiply by themselves. They need a host. That means they need us or any other living cell to multiply. And so that's why they are called acellular. And otherwise, all of others can survive by themselves as well. And there can be mechanisms to inactivate them, and it's a continuous fight between them and any of the higher organism. For example, even viruses can infect or feed over a bacteria. So sometimes bacteria win, sometimes virus wins. So it's always really the hierarchy, and that's how every organism has its own system to protect itself. And for us, we call it immune system, and it is very complex because we have evolved. We are at a really high complexity level of our organization of cells. So once we talk about cells, then I wanted to little bit show how generally cells look like. I'm sure you have also gone through DNA and have seen a lot of different things, but this is just two images, how you might see a cell really in the microscope. This is just different kind of microscopy. We should be actually not colored. That should be more close how our cells would look like. But there are mechanisms, protocols where you can color different parts, for example, some mitochondria, outer membranes of cells, and this is which big vacuole you see is actually nucleus. So that's how you can see the cells, or many other way, very small organelles of cells also you can see. And then if we draw schematically how it looks like. So this is where our DNA is living inside nucleus, protected by a membrane. So that means this is an eukaryotic cell. And from there are various mechanisms, many steps involved by which RNA is getting formed. This is called now transcription. And from RNA, once it comes to protein, then it becomes the translation method. Now, so these are stages, and once you just see where the cell lies in the chart of how to visualize it. So it's really 10 micron. That means 1 mm you can still see in one of the scale, and imagine so many times less. All are decreasing 10 times from here to here. And so imagine how small these particles or other molecules are that we cannot see. But we have developed all the methods to see and recognize all of them. Even small molecules can be seen by very sophisticated machines. That what are their structures, what are the molecules attached to them. So that's how it's the advancement has been done, and we are talking here about animal cell. And within that, when we talk about RNA, so these RNAs are capable of forming proteins. For us, mostly one RNA forms one protein. And this is a different mechanism how prokaryotes work, because they have to accommodate everything with very small information. Is in a small space, but made a lot of information. So they have different mechanisms. And that's how then protein is formed. And protein is really the unit of, like, functional unit of work. So everything runs by protein, mostly. Like, so if you say here, mostly the staining will be done on protein. Okay, then little bit coming about when talking about cell and immune system. This might look complex, but I will just explain a little bit about it, how immune system works in general. So there are many types of cells in our body, and all of those interact with each other. Some of them are really the first line guard, you can say. These are antigen presenting cells. Whenever they see any infection, then they alert different type of T cells, which can recognize only typical very specific non-self substance only. So they are not going to recognize our own protein, but as soon as they see something foreign, they become alert. And then there is a chain of reactions. They can become other type of cell, then they become memory type of cells. They go live at some other place of our body. And it can be also, so each of the cells ultimately, once they recognize, they become active, they try to kill, eliminate that substance, and after that they become memory and get stored in our bodies. And these are the antibodies, what you keep listening about with corona here, we are struggling with. So these are generated by B cells. These are also one of the mechanisms to fight these pathogens. Now, how this I will leave, I will not go into detail, but they all have different mechanisms to either kill the pathogen, to kill the infected cell, and to warn other cells of our body. And that's how actually we also get redness any place, even if we are hurt by something. So all these cells start accumulating there to recognize what is the problem, and that causes swelling and redness. So these are all immune cells which does that to us. You might hear these terms, so cellular, humoral. So whenever antibodies are talked, it's humoral immunity. Whenever any cells are involved, this is cellular immunity. Innate, adaptive, these are different classifications of immune system. For example, these kind of cells which are really capable of fight by themselves are innate. Then these cells need help and a long-lasting memory. So these are adaptive. That's how it's a very superficial view how immune system works and look like. And a little bit what I was talking about antibodies, that's how you can imagine again. So if any virus or a pathogen interacts, or any other immunogen as well, so that means not only the whole pathogen, but whenever even the part of that pathogen is seen by our body, then these are those antigen presenting cells which recognize them first, and then warn other cells or kill those cells, and then these cells warn B cells, and that's they start producing it. So antibodies, that's why are small molecules which circulate in our system. It's not a cell, but sometimes they are also attached to some kind of cells. So they can be of different types, and then these cells just again convert to memory B cells and remain. So that's why some of the for some of the infection we have long-lasting memories. That means we do not need to immunize or face that pathogen again and again for our body, because whenever we will face that pathogen or any part of it, these will become active and take care of it. So that's what scientists try to understand as soon as they develop a vaccine or for any infection, that how long lasting the memory is. So it's not only presence of antibodies, actually these cells matter most out of it. These are checking antibodies in our blood is easy and quick way to know it, but actually the memory cells play all the role. So now these are some of the questions. I'm not going into detail. This is for your curiosity. Go ahead and search for it, that from where these cells are coming from, and then when they become memory, where they are going to sit in our body, what different types of antibodies exist, and how the class switching occur, or it's actually really interesting how they are formed. It's enormous, millions and billions of combination our body is capable of doing and generating antibodies. And that's how so many pathogens existing in the nature, we are protected. So itself is a very interesting mechanism. Then how and why they don't recognize our own protein, they don't start killing us. Then how, and then if you imagine how many numbers of antibodies are circulating, that will be just an imagination actually. And then what happens when these antibodies bind to pathogen? That means how they are really killing the pathogen. So those are some things which you may look for to solve your curiosity. This is TB. I will also not go into detail of it, but it is one of the very clever bacteria existing in nature since millions of years again, maybe since the beginning, maybe before us. And it kills approximately, even within India or worldwide, if I say maybe 10 lakh, 1.2 million or something people, for example, it kills. So before Corona, that was the only infection which was killing most human on Earth. Corona last year changed everything. And it knows how to hide, how to escape those routes which our immune system has derived. And that's how itself is an interesting disease to learn and work on, and also very complicated if somebody wants to make vaccine or drug for this disease. Okay, then this is just giving you overview how what I was saying, that cells have their own way of communicating to themselves, and proteins are mostly the functional unit. So all of these molecules you are seeing written here, they are different types of proteins. Some are recognizing something, some are then from the other part, they signal to other proteins, then they change their form and alert other protein. And that's how the chain continues. And those kind of signaling pathways are well known, but it's a lot to study and find out always in those mechanisms that what other is activating any other molecule. And ultimately then some of the cytokines or some other proteins are generated, and everything turn into those DNA and RNA level that they say, okay, start transcribing this protein, we need something like that. And these all mechanisms are so quick that within minutes you can see the response and ready to act, for example, in cytokine. Okay, so I'm coming to now vaccines, and it's a long history now for vaccine development, and it started mostly with Edward Jenner, who discovered vaccine against smallpox. And that was really interesting, just mostly an observation that people maybe living with cowpox around them were not getting smallpox. And that's how he started believing and testing some experiments that what if that is protecting them. And that was the truth. And this is very crude form of immunizing anyone, that if some person is infected, take some sample of it, give it to other person, and they might develop immunity. It has many its own drawback, and that's why this cannot work and work at large level. That's why this is not a method or way of vaccine development. Although you might have heard this plasma therapy when COVID was going around, that was really trying to save someone at the end, if it's possible. Yeah, and the stories continue. There are many interesting stories around all of them. Louis Pasteur was one of them. Behring was one, Paul Ehrlich was one, and for all of them, there are where they were working, they have mostly formed institutes on their names. So even if you go and search, you will find Pasteur Institute, you will find about, for example, not here, but Robert Koch Institute, who is still focused on tuberculosis, and Robert Koch was also trying to develop vaccine against TB, although he was not successful. But that's how those big scientists, they just dedicated their whole life, and they have helped us to come through this way. So this is still just 2010, and discovery was a little slow actually. Not I wouldn't say slow, but these vaccines really treated most of us a lot, and still are mostly. So we take these vaccines as childhood already. Others are seasonal or needed or changes. So this is, and if we take the vaccine, for example, this is the data from UK, how really you can see the effect of vaccine. This is really for the people. You may go and tell that how vaccines have saved humans. Whenever it was diphtheria vaccine was discovered and introduced, so you can see whatever number of people were dying now, it went to almost zero or decreased after every vaccination. Only some peaks when you see, these are the kind of peaks. Now you also hear about corona vaccine, that this is a wave coming. Because at that wave, either it's our problem from our side, or if that virus changed a little bit, that means the vaccine is not 100% effective, and either vaccine needs to be changed or we are also adopting continuously. So that's how these waves come even after vaccination. But ultimately, that's how we are living. And if you also go back and see history of world about infection and deaths, it was really high number of people were dying, which we don't count now. But there has been very big pandemics all the time. Okay, so now a little bit about how vaccine is generally developed. So a lot of research in the lab, a lot of research in any model, animal model, which is needed because we cannot go directly to human. And also everything cannot be done just on the microscopic in vitro level. And then different trial phases come, 1 to 3. I will little bit tell about it on the next slide. And all these things are monitored by many regulatory agencies. For example, this is the European Medical Association, which is regulating, conducting trial or allowing it within Europe. And then then they allow, okay, that's you can go ahead with this vaccine. So then whoever is the manufacturer, they scale up. And that scaling up involves so much of quality checks that it has. We know it is going to go to the human, and more than any food or anything, these are checked to have purity, to know every component of it. So that's not only scaling up, that's a huge process. Involves many people, a very high sophisticated techniques, and after that, still lifelong almost, these agencies follow safety. Like monitor them, that whenever some cases are occurring, so they investigate if it is because of the vaccine or just a random event. And that's how general time look like. Just a little bit, because you might have heard also these trials, clinical trials during corona pandemic or any vaccine coming, phase 2 result is not there, how much, this and that. So these trials are also very big and actually involves millions of money from people who are making it as well. So first phase is just to know if the vaccine is safe, no side effect is there. They really closely monitor people and very small number of people. Then phase 2, little bit more higher people number, so that now side effects and also the effectiveness of vaccine can be followed. Then phase 3 is a lot bigger, hundreds of thousands of people, where they are also compared if, for example, in 1 year, somebody fell ill and one group was not given anything, which is called placebo, and how many percentage of them fell ill. And that's how it's calculated, that how much percentage the vaccine saved people, or any drug actually as well. Yeah. Then why really corona vaccine was so fast developed? People wonder about it, that no other vaccines were so fast. First of all, this technology is really fast, RNA technology, which was not possible before. Any protein or any other vaccine development is not possible so quickly, and also not possible to change the components so quickly. Plus, it was actually really hard work, because I have seen within BioNTech as well how people were working day and night in shifts, no weekends. And plus, regulatory agencies knew that something big is coming, so they also increased their power. They increased the efficiency, they were reviewing the documentation. They allowed rolling submission that, okay, something has changed, come to us quickly. Otherwise, even going to them actually takes 3-4 months, even taking a date from them and then getting some answer, again some months. So that's how any drug or vaccine development takes years. Those clinical trials from discovery to be able to go to human generally takes a lot of time, which all was saved because of this acceleration phase. And definitely every measure were taken how to accelerate the process of application, and one agency from one country was considering the data from other country. So this I wouldn't spend too much time. Maybe I'm almost towards the end of my presentation as well and time. So this is to show how different types of vaccine could look like, what we generally take in our daily life. So for example, BCG vaccine here is against tuberculosis. That's a live attenuated bacteria. And different types of exactly vaccine exist. We generally are taking as childhood hepatitis B, for example, subunit vaccine. This is now SARS-CoV-2, which is mostly DNA or RNA-based vaccine. Some are also, for example, within India, when you are taking Covaxin, this is attenuated virus, and that's also a very good mechanism or the way of making vaccine. That would be actually the best if it works, because it generally involves whole organism. That means our immune system has many components to recognize. But the problem is they are risky. They are very risky because if something a batch or a batch of vaccine, any one virus or any one bacteria remain deactivated, then it can cause disease, and it has its own limitation. It cannot be given to all people, some specific people, and it can cause more side effects. So that's why so many techniques are on the way and have been developed to to tackle different problems. So exactly, so here we are at RNA vaccine, where it's very simple. It's generally some RNA molecules inside, which is covered by lipid molecules and some other solutes, some salts, etc. And everything is in some kind of buffer solution, which is making it stable. Or actually the molecule itself, this lipid nanoparticle could be one of the carrier of this. It can be also a lot of different carriers. The whole point of having this concept, or how to say, it's like how it is stable and how it is formed is very, very basic chemistry. It's like water, liking molecules like to be together. If some molecules are not liking water, then they try to revert themselves. And if you try to imagine two of the molecules which are not liking water, and water is on both sides, then they like to try to face each other. And once they are facing each other, then if some molecules or particles are within the solution, they will be trapped inside them. So that's one of the mechanisms how RNA can remain inside. So you can search if there are different, could be different carriers, lipid-based, carbohydrate-based, in different buffers, and that's just the basic mechanism how they are carried, and that's how they are also protected. Otherwise, you might have heard, if you just in our skin, we have so many of enzymes which can just destroy any RNA or DNA even by touch, or even if our dead skin fall over them. So that's how the delicate these genetic materials are, and they cannot exist outside the cell, and that's why it needs to be having the carrier, which is then delivered. And they also have different properties that they can target and get to them, to different cells. And these all, for example, these all could be different ways how they can be manipulated. First, against which target these RNAs are generated. if it is host or pathogen, and that can define if it is a therapeutic vaccine or a prophylactic vaccine. For example, if I want to develop immunity against coronavirus, I will form this RNA against some component of coronavirus and immunize me. Then I will develop that protein, which is kind of will be present in coronavirus, and my immune cells will recognize it and remember it. And if, for example, for a cancer therapy, if RNA vaccine has to be used, that's a therapy, and then there is no pathogen involved. But then there are different new epitopes or different targets of our own changes of cells, which are recognized to be encoded by the RNA. And when our cells recognize and see them more, they try to eliminate that component. So technically eliminating the cancer cells, not so which are little changed. So very fine tuning can be done based on backbone and carrier type and also route of vaccination, so that these aims are achieved. And that's how different types of immune responses are generated, and we get different outcome as well. So this is new field. A lot of scope is there. Technologies are advancing. For example, very little idea about this one of the paper, which you can also see. This is one of the people from biotech community or related, that how once you inject, different types of cells are taking those lipid enclosed RNAs. Yeah, these are some tissue resident cells, and also then those information are passed to our lymph node, and that's how it's different cells which are taking and activated or identifying different features, and then there will be changes on those cells, and there will be other changes following up. So it's a huge mechanism. A lot has been understood, and many things as soon as advances of technologies can be understood. That's where a lot of the scope of technological advancement is, and I hope you will contribute to that. So that's all from my side.
Thank you so much, Neha. I mean, am I audible? Yeah, great.
Yeah.
So I don't know how much people understood what Neha has said. Like, she really took from the basics of immunology to the cutting edge research, what she is doing. I mean, why I was super excited when Neha said yes to this talk is that she is in the field and she is developing vaccines. She's not like somebody else who is preaching about the technology or methodology, but she's the one developing things. And BioNTech, with the help of Pfizer, they made the first corona vaccine of the world. So that means, like, we are so privileged to have you, Neha, here. So it's not easy to get people like you, but thank you so much for giving the talk.
I'm happy to be here, Jagdish. All my pleasure.
Sure.
Yeah, thank you, ma'am. Any questions from the participants? They can type in the chat box, or they can raise their hand so that we can unmute you.
So
It's
Neha, there's a comment in the message, like, immunology quite difficult to understand, but good lecture, ma'am. So do you want to comment?
Thanks, Jagdish. Yeah, so Rajesh, it's not possible to get immunology in half an hour. You have to read those books and understand a lot. My aim was to let you know that this is a big field. A lot of scope is there.
Yes, ma'am, we have worked with the tuberculosis also. So that is also a complicated immune response there. So it is still growing every day. Yes, ma'am. So, yeah.
Mamata raised a hand, right? R. Mamata, DGT Bioscience.
Shiva, if you're there, please unmute Mamata.
I did, ma'am.
There's Pranav also.
Yeah, good afternoon, madam. So I have a doubt regarding this Covishield. So many are okay safe with Covishield, but some of the what do I say, they got victimized because of what do I say, clotting in a brain or blood clots they are finding. So what is the reason behind it, madam?
This I cannot go into detail. Yeah, I also heard that those are concerns. Any vaccine can come with known unknown side effects, and something like that must have happened with them.
Yeah.
And that's how it is. It is so. This is what actually regulatory agencies do. They follow these, and if they are seeing some side effects are there, they go back and check if it is from that vaccine, and then they try to say don't. So it is allowed only for this age group. It is only allowed this much time gap, and that's how these regulations are formed. So even though those news or some events were there, but this is in the field, people are taking it, and as soon as everyone and doctor and patients are following what is recommended by agencies.
Yeah, actually, I heard just somebody are facing this risk, but I don't know what is the main reason behind it. So that's what I pose this question. Another one, madam. What about these memory cells regarding Covaxin? They are not at all. So many are telling that it will be memorized only 6 months only. It will work only for 6 months. Why can't we develop the permanent vaccine once we get it, and further we'll be safe from different coronaviruses?
It's not only coronavirus, that is something really our immune system dependent. That's not on vaccine dependent, because even after having infection virus itself, that memory is fading. That's the news more important. So don't look it from the vaccine perspective. Look, because something our immune system cannot remember,
Yeah.
that means we need boost. Either by take again vaccine, get or get infected. So either way, you have to remind yourself that so that's there is no answer to that, because it's our system, and there are so many. For example, tetanus. Why it's said to take after some time, because after some time we don't remember and we don't want to get tetanus infection. So that's why the vaccine is recommended. And whenever we cut ourselves, we go to doctor. I missed it, so I should have it.
Last, madam. Last but not least, last. What when we can see the what I say end of this virus? Actually, it is getting into the different humans, and it is reconstructing. Recombinations are coming, new variations, strains are coming out. Does it comes in with heredity, or does a herd immunity, or it is depends upon individual food habit, diet, exercise, etc.? What can you say, ma'am?
Yeah, that's something big to comment. I will not talk about herd immunity, but anything which comes for us from nature, it finds a balance. So whatever virus changes, we are also changing, and plus our technologies are helping us. So I would say we can be patient, follow regulations, and then we will overcome it. It's better already since last year, right?
Yeah. Right, madam. Thank you so much for the valuable answers. Thank you, madam.
Good afternoon, ma'am. I'm Mamta, TGT Bioscience from TSWRS Valanda School. So, ma'am, I have a doubt, ma'am. So, thank you so much, Neha, ma'am. You explained very nicely the introduction of the immunity, how they are going to be boosted in our body, and hypersensitivity and the types, everything. Ma'am, here I have a doubt that during the preparation of the vaccines, so which method commonly we follow different methods, but as a discovery, during the discovery, ma'am, which method you followed for the making of a vaccine, ma'am, co-vaccine?
So a vaccine, making vaccine. So it is first, what should be the target? Like if we know the spike protein or anything, any of the component from vaccine or pathogen you identify should be recognizable as soon as that pathogen enter our body. So the target should, so first step is always identification of target, because if we want response to A, first thing which is exposed to our body, and then we should not make a vaccine against B, because then we are already sick as soon as that pathogen enters. So identify the target, then those method of vaccine developments are already established. For example, for us also, it was I should not speak from BioNTech side because anyway I joined late. They had all these methods established. So once you know the target, then you try to see if it is working in the lab, in animal system, and then you can say that, okay, with our technology and this target, this is working. And now that will go forward.
Ma'am, I have one more doubt, ma'am. So recently, I read in a newspaper that whoever individuals post patients of corona, they are taking vaccines, ma'am. After taking the vaccines, their lifespan going to be reduced up to 2 years, ma'am. Is it true or the fake information, ma'am?
This must be fake. This much I can just hear and tell. Why vaccine will reduce the lifespan? That's not the case.
Maybe it increases.
Okay, thank you so much, ma'am.
Yeah.
Right. Thank you so much, ma'am. But because we are falling short of time, I will have to cut down on the question answer round.
Definitely.
Thank you very much for joining us, and I hope that we can further be associated with you as well.
Yeah, sure. Thanks, you all.
Ma'am, you can take over.
Right. But Neha, feel free to be around and maybe answer questions in the chat if you have time. Yeah.
I will be for some time here, and then for sure. Otherwise, yeah, you are also capable of answering.
Sure.
Thanks, Venkatesh.
Yeah. Bye.
So after such a wonderful session, we have with us Professor Anil Sharma, who is going to give a talk about the CRISPR revolution. So Professor Anil is actually a molecular geneticist with expertise in genome engineering and editing. Am I audible?
Yes, Sumati, can I take over?
Yeah, sure, ma'am. Sure.
Okay, fine. So, fine. Good afternoon, all. This is Dr. Purnima from the organizing team DNA 150 Years. Revolution is a part of civilization. It indicates positive learning culture. So we are progressing and heading to the era of gene editing to answer the queries of human needs. Gene editing is a group of technologies that give scientists the ability to change an organism's DNA. This facilitates the genetic material to be added, removed, or altered at specific location using CRISPR-Cas9. Ladies and gentlemen, please help me in welcoming the key personality who is hailing from the University of Alabama, Department of Biology in 2018, experimental biologist, expertise in genetics, cell biology, developmental biology, CRISPR technology to understand the gene function and disease, developed course-based undergraduate research, cure, extensive work in the experimental design, demonstrated success in research, education, outreach programs, product development, market analysis, and genome editing. We now welcome Professor Anil Kumar Chella. Very good afternoon to you, sir. The platform is open to you. Thank you.
Thank you very much. Thanks a lot. Am I reasonably audible? Can you hear my voice okay?
Yes, sir.
Yes.
Thank you. Give me 1 second. I will be just trying to set up my slides. Can you see the slide at the moment?
Yes, sir.
Thank you. Thanks again for the invitation. It's a delight to be part of this group right now, and I'll share a little bit of my experience. So, well, I will leave enough room towards the end to share my personal experiences with the technology. But from my understanding, this was more like an overview because of a variety of topics. So I thought I will take you through the journey of this particular technology. 1 second. Okay, sorry. Yeah. So I gave the title of for this talk, the CRISPR revolution from a bacterial immunity and precision gene editing. And I think I'm very fortunate that Neha actually gave a talk about immunity, the way we generally know about. So this is a story of how bacterial immunity kind of works a little bit, and then how it has given us an amazing tool. In fact, a bunch of tools that has revolutionized the biotech industry to say the least. So the way I'm going to start is one of the focus of genetics is to understand gene function, and eventually came in the last 20-25 years, it has become our endeavor to understand genome function. So initially, for the last, since prior to Mendel's time, people have been interested, of course, in understanding what genes are, not in that particular in the form. So there was an understanding of inheritance, and that there's something organic about it. There is the intelligence behind inheritance is within the cells, right? So from the late 1800s, this understanding was there, and now the goal was to find out how that happens. So as all of you, and most of these characters that I'm going to talk about, at least in the next couple of slides, you are aware of it. So Mendel used or exploited natural variation. So everybody's aware of the whole tall and the dwarf plants and the pea pods and the like. So he used naturally existing variation as his experimental tool to understand, and controlled pollination to understand how inheritance happens. And early in the 20th century, Thomas Hunt Morgan, and again, these are all textbook names, they used spontaneous mutations. They were able to identify spontaneous mutations, and they used them as sources to understand gene function. His own team member, Hermann Muller, used ionizing radiation to induce random mutations. And then there were technologies or tools that were developed or available, which allowed people to induce random mutations by chemicals. So while Muller's work was related to inducing mutations by ionizing radiation, you have Orberg. She induced random mutations by chemical agents. Well, she's known for that work, but many others also contributed to the work. And then the famous Barbara McClintock's work using mobile genetic elements. So the common theme of this particular slide is that we have been exploiting or looking for tools that enable us to understand what genes are and what they function as. So this is just as a little bit of a background, how these things have been helping us. Using these tools, the tools I've already mentioned, helped us understand forward genetics, or what came to be known as forward genetics, where you disturb the function of a gene internally, but which essentially shows up as a phenotype. So trying to understand the phenotype and the underlying genetic factors was the endeavor, or is still the endeavor, of forward genetic screens. So using some of these tools, large-scale mutagenesis screens were done, as I mentioned, with all these tools to discover gene function. So that is a backdrop. So while that all was happening, we were understanding a lot about the cellular basis and the molecular basis of how life works. And bacteria turn out to be a very important participants in this whole journey. And let's start the journey with the discovery, right? So Anthony von Leeuwenhoek, again, textbook figure, in the 1600s, late 1600s, discovered a whole bunch of small things under the microscope, and one of them were bacteria. So this is a handprint of an 8-year-old, and you can see the diversity of microbes on a petri dish, right? So there's so much diversity, and this actually tells you a story of plentiness and plunder. When there is prosperity, you can see basically what looks like a very blank petri dish is actually a lawn of bacteria. And on the right side are plaques. Some of you might already recognize. These are viruses which attack bacteria. So wherever there is prosperity, that does invite plunder. And of course, that is very anthropomorphizing the whole thing. But the story is not as simple. So there is plentifulness and the bacteria are happy and they created a lawn, but that invites plunder by the viruses. So think about it. It's not very different from when you have resources or people to protect, you create forts. And so this is an example, I believe, of the Jhansi fort. And once you have something like this, there's a reason why this came about. It is to protect resources and people, but that also invites enemies, right? So we know the story. So there were a lot of people who were trying to plunder any of these forts, and it is not going to be taken lying down, right? So there's a lot of defense happening. So the outside wall of the fort is your first defense against invasion. You can actually weave an entire story where, how does an invader trying to attack this fort, what kinds of strategies would they adopt, and what kinds of strategies do the people inside the fort adopt to thwart the invasion, right? So I'm assuming that you can easily imagine a nice Bollywood movie, which, with awesome graphics these days, in terms of this whole arms battle and how the strategies keep developing. So not very different from that. Anyway, to give you a backdrop of this whole thing. So as a continuation, what's the whole idea about the plunder? It turns out these are bacteriophages, I already mentioned, and there's a nice, interesting relevance. So in 1896, Ernest Hankin reported something in the waters of Ganges and Yamuna rivers in India. They had a marked antibacterial action against cholera. And so it doesn't surprise us actually that people, as a what turns out to be probably a very superficial ritual right now, might have had an extremely, in fact, there is enough evidence, has probably had a very interesting scientific basis, which is, cholera was a very common thing even until like 60, 70 years ago in India, and not completely unknown even in current India, for example, or many of the developing nations. So what happened was when people took a dip in the Ganges or so-called, very sacred rivers, is potentially the load of bacteriophages. Anyway, maybe I'm giving you the punchline already. So in 1915, British bacteriologist Frederick Thwart discovered a small agent that infected and killed bacteria. And this is generally considered the hero of this whole story, Felix Derel, a French-Canadian microbiologist. So he independently, working at the Pasteur Institute in Paris, announced on 1917 that he had discovered, and in his words, an invisible antagonistic microbe of the dysentery bacillus. In a flash, I had understood what caused my clear spots was in fact an invisible microbe, a virus parasitic on bacteria. Derel called the virus bacteriophage, a bacteria eater, from the Greek phage meaning to devour. And this is probably, again, textbook knowledge. So essentially, it turns out, any of these like Ganges has a heavy load of bacteriophages, which could potentially infect the pathogenic bacteria. And that's probably why people got cured. And therefore, they attributed the divinity to the river. But the underlying idea is probably the bacteriophages. And some of you might already know that there's an entire movement to bring in bacteriophages as antibacterial agents to fight bacterial diseases. Anyway, so he also recorded a dramatic account of a man suffering from dysentery who was restored to good health by bacteriophages. So again, this is just a cartoon of how this might be happening. The phages are infecting this fortress of the bacteria, and this is the outcome of that infection. You have all these viral particles getting out, and this is your classic lysogeny, if I have to use a textbook term. So let's ask a question and say, like, if you were the bacterial cell, how would you fight the invading phage? And some of you are already aware of these things. You will probably say, well, we know how the bacteria has evolved strategies, right? But I'll give you like 10 seconds to think about how the bacterial cell might be fighting the phage. Okay. In a normal circumstance, we would have had a nice conversation, but since it's online and lots of, go ahead. If anybody wants to contribute,
There are two answers, Professor Challa, in the chat. So
good.
One answer is plasmids. One answer is plasmids, and the second one is using CRISPR-Cas9 mechanism, defense mechanism. Another one.
So these are all very, very difficult words, right? So I really don't know what plasmid. It's a very loaded word. I really don't know what plasmids mean, and CRISPR-Cas and nucleases. These are big words, but in general, but I think the idea behind that, I'm guessing, is that you want to really kill or you want to destroy what is the real agent that is causing the infection. And it turns out in the bacteriophages, it's the DNA of the bacteriophages. So any mechanism that allows to destroy that particular invading DNA, be it plasmids, be it viral, any pathogenic plasmid for that matter, you could potentially, that needs to be destroyed, right? So once you have an invading, so it's not the protein, it's not anything else, it is the key element, that is the nucleic acid. So that is the agent that is causing havoc inside the cell. So it turns out, evolutionarily, as we mentioned, any kinds of arms race is, it's a two-way street. It's not just that if the phage in reality was so successful in creating a situation where it's able to eat up every bacteria, then the story would have ended right there, right? Because if a completely effective phage would have completely destroyed all bacteria, at least of a given strain, but that did not happen. It turns out bacteria have evolved a system where they can actually digest, basically destroy the invading DNA. And it turns out, and again, this is what is classically known as the restriction modification system. We'll not get into the details, but it's almost like there is a magic samurai sword that the bacteria has gained, and then it is able to kill off or basically chop off the invading DNA. But the problem is that same samurai sword has the tendency to probably destroy its own DNA, because DNA by its nature is not distinct between phages and bacteria, right? So it turns out, along with the samurai sword, it was also blessed with a shield which can protect its own DNA that is not available to the phages. So that is a modification system. And that is, I mean, again, over 20, 30 years of work, people discovered that it's a methylation or the modification system that methylates the host DNA while leaving the invading DNA without any protection. And the restriction enzyme, these are basically endonucleases. They are like the samurai swords, which can break down, which can cleave the invading viral, the phage DNA, and the native host DNA is being protected by the methylases or the modification system. So this was discovered in the 1950s, and as most of you know, that this has actually led to an entire revolution in the biotech industry, right? So this is a birth of modern biotech industry. It's a very, very famous meeting of Herb Boyer, who was doing some very, very key experiments in molecular biology using these enzymes. And Robert Swanson, who was an investor, who actually said, okay, can we have a quick meeting? I'd like to talk about the potential of, capitalizing these enzymes. And he said, like, I don't have time for all this stuff, so maybe I'll give you 30 minutes. And that 30 minutes turned out to be a 3-hour meeting, which was which led to the birth of Genentech, the first biotech company in the modern sense. So this is an old cartoon. This is something, how to turn a bacteria into a hormone factory. The key thing here is that the plasmids, which are, typically by that time in the 1970s, they were able to access these DNA, but restriction enzymes, which were able to open up the plasmid, and now plasmids were the vehicles to take any payload, anything, any piece of DNA that you are interested in, to make whatever, to make bacteria as factories to produce anything that we are interested in. So that's, basically gave rise to, as I said, the first revolution. So what that did as a technology is these site-specific bacterial endonucleases allowed us to understand genes and genomes like never before. So we were able to really access physically pieces of DNA and how information is encoded. So I'm really cutting through a lot of history. In addition to that, when you brought in DNA sequencing, the ability to actually read every base in a given stretch of DNA, though that combination of restriction endonucleases, plasmids, and DNA sequencing technologies, and the whole lot of chemistry behind it, led the way to something called reverse genetics, where you actually have the sequence available, because you can actually now sequence DNA, And then once you know that particular DNA, you can actually start disturbing that gene or manipulating that gene and asking what would happen to the resulting phenotype, either you when you basically manipulate genes, right? So again, this is at a very the resolution is not so great, and you're talking about you can't really change mammalian systems or even complex eukaryotic systems, but potentially are a lot of viral and bacterial systems were now being manipulated. So this led to the reverse genetics revolution. And I'm trying to get to the idea of remember the underlying idea is still how do we understand gene function. So this is in the 90s. So these three gentlemen, I mean, this all the work happened between the mid 80s to onwards. Mario Capecci, Sir Martin Evans, and Oliver Smithies, they were recognized for their work for introducing specific gene modifications in mice by the use of embryonic stem cells. So now they brought in a new technology of embryonic stem cells, and then they were now able to actually do specific gene modifications in a mammalian cell. And I'll quickly go over this. The idea is not to remember every detail of this, but the idea is they were able to culture embryonic stem cells. This is again in the late early 80s and onwards. Once you have ES cells, you could actually create using plasmids. You could actually create a rare cell which with a modification that we are interested. So this is the technology that was facilitated by restriction endonucleases, and I'm not going into the details. And then once you have the rare target cell, you can actually select those cells and put them in mice, and you can give rise to a modified animal. And this was a phenomenal technology. And the reasons for the success of gene targeting in mice was the availability and access to embryonic stem cells, homologous recombination with a donor DNA molecule, which is the byproduct or the product of recombinant DNA technology, the ability to select positive ES cell clones in tissue culture, amplification and introduction of ES cells into host embryos, that is by a process called blastolar transplantation, and screening for germline transmission. So if I go back, so you can actually screen for those animals that are modified already. But this was a long process, and there were a lot of hurdles to go through. This also had, while this was possible in mice, there are species limitations to embryonic stem cells. As we mentioned already, the homologous recombination event is very rare. This whole process is laborious and time consuming. And the low frequency of achieving this modified progeny added to the cost. So in even today, if you want to go through this technology, you'd spend anywhere from $7,000 to $10,000 US dollars to create one of these animals. But the key thing is, if you are able to achieve recombination of sorts, I mean, if you're able to manipulate a piece of DNA on your call, then it's a lot more easy. So in the mid 80s, Maria Jassen's lab and a few other labs, Jim Haber's lab, for example, they found out that inducing targeted double-strand breaks in host chromosomes stimulates recombination at the locus several orders of magnitude. So what is naturally a very rare event can be enhanced or increased when you have a targeted double-strand break. So now once we knew this, the holy grail was, how do you target a specific site in the entire genome? So here is a question for you. I will give you 30 seconds to generally think about it. And I'm going to get some water to drink. My throat is getting parched. So I'll take that 30 seconds for you to think, to get some water. How do you think we can experimentally induce double-strand breaks in a genome? And the condition I'm going to put is, you cannot use big terms like that I don't understand, right? So you got to use conceptual ideas to tell me. You can put it in the chat box, and in about 30 seconds, I'm going to, if anybody wants to volunteer and say it out loud, that's fine too. So think for 30 seconds and tell me in very simple human, any layperson should be able to understand, how do you think we can induce experimentally double-strand breaks? What kinds of conditions, what parameters have to be met in order to achieve site-specific, well, I kind of gave you a clue, double-strand breaks. So give me 30 seconds and I'll be right back. Okay, I see some words again. Restriction enzymes. It's an interesting answer, but if you can think about it, if you want to send a restriction enzymes, let me check the chat again. So there's one answer was restriction enzymes, gene silencing, restriction enzymes. So here's the problem with restriction enzymes, right? That's like a samurai sword. So if the bacteria are blessed with the shield that can protect their own genome, correct? So if you actually send in a restriction, if you're able to send in a restriction endonuclease into a, let's say, a mouse cell or a human cell, our genomes are not endowed with one of those shields. So how can you reach specificity? But the thinking is in the right direction, though, because a phenomenal discovery by, incidentally, an Indian scientist who's not very well celebrated comes to our rescue. So anyway, turns out what we need is two things in terms of conditions or parameters. In order to create sequence specific, very specific things, you need to have a machine, some kind of a bio nano machine, which has specificity and efficiency, which is it needs to be able to find a particular sequence in the large genome and then be able to actually create a double strand break, right? Because if it destroys anything else, then there's a problem. You are destroying information. So you need to have a very precise double strand break without loss of any information. And it has to be at a very specific site. So what kind of a bio nanobot should we build or can be available if you want to look for it, which has both the specificity and efficiency? So targeted gene editing by designer nucleases can be, you can achieve sequence recognition by specific binding, right? So one of the things people discovered was that proteins can be designed. In fact, all cells, all living cells actually have proteins which have sequence specificity. Think about the transcription factors, for example. They can actually bind to very specific sequences, and they have these domains, and we'll come to that very soon. So one of the first efforts to create these double strand breaks was by creating designer nucleases where proteins will be our recognition domains. So this is a very classic thing. This is the beginning of a phenomenal discovery / technology. Srinivasan Chandrasekharan at Johns Hopkins was the pioneer in this. So what he did was, and the thinking was right, if you took the cleavage domain of a restriction endonuclease, and fortunately there are such enzymes where the cleavage domain can be separated out from the recognition domain, and then you use DNA binding motifs, which are typically zinc finger monomers. So binding of two zinc finger nuclease monomers to two specific sites is required for effective double strand cleavage. So this kind of work started off where you could create zinc finger nucleases where murine transcription factors or human transcription factors, which have zinc finger domains, could now be joined or fused with non-specific DNA cleavage domains of bacterial origin. And one of the enzymes was FOC1, and they created zinc finger nucleases where the DNA binding domain composed of zinc finger motifs fused to the FOC1 nuclease to initiate targeted breaks. Here, the resolution of a zinc finger motif was 3 nucleotides. So if you actually had a sequence of 4 to 6 zinc finger domains, DNA binding domains, you could achieve a sequence specificity of 18, right? So if it is 4 times 3 is 12. The possibility of finding a 12-base sequence in a genome is relatively small. If you're able to achieve 18 or 24 by increasing the number of these domains, you can actually reach much more greater precision or specificity in the genome. So you bring 2 of these DNA binding, these zinc finger nucleases, and then you can create a double strand break, right? Now the problem with these is the resolution is a little poor, as in for every 3 nucleotides, you need to create a new domain, a zinc finger domain. So even today, while people use the zinc finger nucleases, it's a lot more work because you have to identify efficiently cutting or identifying the zinc finger domains. But once you are able to identify a very specific domain, it's probably not very difficult to create this artificial nanobot, which is the zinc finger nuclease. So even today, it's actually very expensive to create zinc finger nucleases. And at the helm of it, in 2003, to create one set of zinc finger nucleases with all the work was costed about $30,000 in the United States, right? So that was not economically feasible. Now, the advantage of while this was happening, I'll come to that later. I can't tell you both the stories at the same time. But while that was happening, we were also trying to figure out, okay, if we have double-strand breaks, how can we change things, right? So this was not only allowing us to try to disrupt genes, but also giving us an access to the genome in a very specific fashion, so that where we can probably even protect or change or modify the genome from a disease state to a normal state. So that's kind of a cue I'm giving to where we want to go later on. So anyway, zinc finger nucleases are now created, and now we actually have a tool which can allow us to go into a specific region in the genome. I just noticed that we are already 40 minutes and I have only 15 minutes. So I'm going to run through because we haven't even touched CRISPR, which I promised. So the long story is then TAL effector nucleases or talons were discovered. That's an interesting story. But even here, proteins are the ones which are the recognition domains. So I'll skip this stuff and then we'll go to CRISPR. And that's a little bit of background. And I think I will be able to do justice by giving you this background and also give you a small taste of what's happening. So 2005-ish, or slightly before that, this was crystallizing. The arms race was not just very passive. That is, it's not just the restriction endonuclease system where it's just waiting with a samurai sword, but the system already had an adaptive immunity, right? So CRISPR-Cas9 system, it turns out, is able to not only just randomly cleave the pieces of invading DNA, but can recognize, store that information, and unleash an attack in future infections. So it has three phases: adaptation, maturation, and interference. Adaptation is where the bacterial system, is able to capture a piece of the viral genome and place it in its own genome. So this is like some kind of a bio IDing system, right? So you are basically recognizing, and it's a database. It's a database which had these are all they found out by doing genome analysis that they found these regular sequences. There's a repeat sequence everywhere, and they're always clustered, and they're regular. They're not irregular, and they're palindromic. They're short, and therefore they called it CRISPR. That is clustered, regularly interspaced, short palindromic repeats. So CRISPR array is essentially a database of stolen viral DNA. But they did not know that. So they actually said, okay, these spacers don't make sense, well, but maybe they seem to be interesting. And later on, they found out that the spacers, each spacer actually is a viral sequence. And they were right next to this CRISPR array, where some sequences which are associated with this always, in any species that they saw, they started seeing CRISPR associated sequences. They coded for proteins. So these things, this is adaptation. So when a particular sequence, viral sequence enters the bacterial cell, the CRISPR system is able to cleave off a piece of viral DNA and integrate it into its own CRISPR locus. Once it is there, there's another system within that larger system. It's able to mature. So expression and maturation happens where these pieces of RNA are sent out, and these pieces of RNA, in conjunction with a relatively non-specific nuclease protein, this team, the ribonucleic protein complex, can now become a functional machine which can go recognize viral DNA and create double-strand breaks, and therefore inactivate those viruses. So this is interference. This is the actual action of killing or destroying the invading RNA. So there are lots of class 1 and class 2. I'll not belabor because there's some interesting stuff I'd like to say. But suffice to say that, especially in this one case, we are going to specifically talk from now onwards about a very well-characterized system from Streptococcus pyogenes. So it's called Streptococcus pyogenes SPY, so SPY and Cas system, SPY-Cas system. It turns out there is a very, very interesting grammar that this machine understands. And the beauty of this whole thing is that this is RNA-guided nuclease. So RNA is the recognition domain. So until the zinc finger nucleases and the talons, it was the proteins which were recognizing a particular sequence. But in this case, now it's the RNA. And as you all can imagine, complementarity is an amazing thing between bases that can give you extraordinary specificity that cannot be achieved by proteins so easily. It can be achieved, but the engineering that is required is a lot more tedious. So long story short, these proteins, the Cas9 protein has two nuclease domains, the HNH domain and the RAV-C domain. These are like two pieces of, two arms of a scissors. And upon recognition of the RNA to the targeting DNA, you are able to recognize, and then it can create a double strand break. And there are again two kinds. I'm going to talk mostly about spike Cas9, but here you see that there is a specific recognition sequence, which is a low-level recognition, which is called a PAM. And upon that, this orange sequence is the target sequence, the CRISPR RNA. Once the recognition happens, there's a double strand break at the third base pair from this first recognition. I might not have given you a very clear picture, but suffice to say that this is a machine which uses RNA to find its complementary sequence in the genome, wherever it is, and then create a double strand break. So this is the system, and this cleavage is happening, and this can be now used to do some manipulations. So it turns out already, I mentioned this briefly, but homology-directed repair is a major double strand break repair pathway, and I mentioned this already. So Madhuri has a question, or she raised her hand. Is it a question or a comment? Okay, maybe I'll just take it as a chance thing. So, okay. So once the double strand break, what happens? That's it. That's the end of the CRISPR-Cas9 system, because the bacteria is able to achieve what it wants, which is to thwart the viral invasion, correct? Now, we are able to now, like the way we use restriction endonucleases, we were able to now use this system to say, well, now we have a nice tool to create a double strand break. So how do we use this to either understand gene function or to maybe even change gene sequences as we please? So mammalian cells have been presumed to repair potentially large. I already mentioned they do not require homology break site. This contrast to, I'll skip this because I already mentioned these points, and I'm sorry about this. All that text I showed you earlier is going to be summarized here. Whether it is a zinc finger nucleus cut or a talon cut or a CRISPR double strand break, there are two paths that the cell can take. One is relatively non-specific, but can be precise or imprecise repair system called the non-homologous end joining. There's not much intelligence here, but there's a joining of the two ends. If you're lucky, the joining is precise, or in some events, it can lead to insertions and deletions. Or if you have a homologous repair template, that can be used in order to direct this repair through a repair system called the homology-directed repair, right? So either through non-homologous end joining or HRD, now you can repair. If you can create, so we'll go to this. If you take the normal wild-type CRISPR-Cas9 system, because of the possibility of creating indels, you can now create mutations at will, or through homology-driven repair, you can probably create precise gene editing. So this is using wild-type Cas9, right? When you create indels, you can actually create mutate. That is a mutation. And therefore, if you have an unknown gene of unknown function, using the idea of reverse genetics, you can manipulate it and ask what goes wrong in a phenotype and attribute that phenotype or the behavior of a cell to the loss of that particular gene. It is a very reductionist approach, but it is a very, very, very precise tool that enables us to understand gene function. Now, the advantage you can probably immediately imagine about precise gene editing, because now imagine this is sickle cell anemia DNA, and if you have a guide RNA that can precisely locate that particular mutated sequence using homology-driven repair, you can actually correct that particular mutation. And that is the excitement with that this particular, and all of this is purely by using RNA as a recognition factor. So just with this, you can see the dates somewhere here, 2017, 2015, 2016. So you can improve crops precisely by CRISPR-Cas. You can engineer pigs for eliminating endogenous retroviruses in a step towards xenotransplantation. It's actually affecting agriculture. So first, clonal rice seeds are an agricultural dream come true. And then that gave rise to the possibility of, well, can this now mean something to humanity? And this is The Economist from 2015 already. They're talking about the prospect of genetic enhancement and editing humanity, right? So can you now recognize, if you have a sequence that is for high IQ, can you modify it to create high IQ, have no baldness, perfect pitch? So is this giving rise to, designer babies? That was the problem. Well, that was not left as a theory for a long, like it didn't take long. By 2018, November, JK, as he's popularly known, He Jiankyu, he not only made the first set of CRISPR babies, but the second pregnancy was already confirmed. Now, at least according to the Chinese sources, he's serving a prison sentence because of a lot of issues. But the bottom line is he intended, and there's still a hope that whether we go through germline editing or if we go through somatic editing, this again, it's like any piece of technology, right? So the technology is there. How are we going to use it? What are the ramifications of it, socially, ethically, legally, and all that kind of stuff, right? So I'll quickly go over, because I at least want to have about 5 minutes for questions. So I don't want to go beyond my time. So now I just want to give you what can be done with this system. So it turns out CRISPR-Cas9 is giving rise to it's almost like a rocket and a payload system. So you have something like a nanobot. Let's imagine something akin to a drone, right? So a drone was probably militarily made for bombing enemy sites. But what if you disable that bombing capability of the drone, but now add different things, right? If you put a camera instead of a bombing device, you can image things. So you can, and it was indeed done when they made a GFP fusion with dead Cas9. A dead Cas9 is a Cas9 protein unable to create double-strand breaks. You can now use this to image specific DNA loci in living cells. Or you can actually fuse proteins like VP64, P65, which are basically transcription activators, or CRAB domains, which are transcription repressors. So instead of cutting DNA and disabling genes sequences, you can actually enable, activate or inhibit gene expression purely by using this particular drone, this particular CRISPR-Cas system. Again, using the dead Cas9 as a rocket and any of these other modifying proteins as payloads. Genome editing can also be done by using histone or DNA methylases. Modifying enzymes can be also attached. And this allows us to go in and query or reach out to specific sequences in the genome. And then the payload enzymes can do the modifications. And again, base editing, I'll spend a little more time on, maybe 1 minute before we close soon. Some amazing protein engineering effort led to using these base edit, these CRISPR-Cas system, and they evolved proteins which can now not cut, but truly edit. So you can actually just change an existing C to a T, or an A to a G, or G to an A for that matter, right? So you call them adenine base editors, or just cytosine deaminases. Base editors, essentially either cytosine base editors or adenine base editors. And the last thing and the latest thing, which is the craze of the CRISPR field, is prime editing, where they engineered Cas9 protein. Now it's not just a dead Cas9, but a nickase, and there are reasons for it. And if I have time, I'm more than happy to answer those questions. So they were able to fuse reverse transcriptase and not only give a guide RNA as a targeting sequence, but also a piece of RNA that can specifically repair that particular nicked sequence that the nickase, or the slightly modified Cas9 protein, which can create a single strand break, can achieve. So once a single strand break has been achieved, they've engineered this prime editors, which can actually bring in a sequence that needs to be modified right in that proximity. So this is a big craze right now, and they're trying to improve this. And there are about 3 or 4 companies already trying to do the beta testing or some, not phase 1 quite, but preclinical work to see if we can use this technology to correct any genetic errors. So because we are talking about CRISPR technology, even though I said that we are interested in understanding gene function, I cannot stop from saying that I have to tell you that and some of you might already be aware that CRISPR is also being used in diagnosis, because not only are CRISPR systems able to detect DNA templates, but can recognize RNA templates. So if you have RNA viruses like the SARS-CoV-2, for example, you have a CRISPR-Cas system called the Cas13a, which can be used, and using a different set of technologies. These are again molecular biology, chemistry, like biochemistry stuff. You can actually use CRISPR even for diagnostics. And this is, I'm just spattering of different ideas that have been exploited using CRISPR technology. So just to give you an idea of the commercial impact, the CRISPR, there are at least 3 leading companies: CRISPR Therapeutics, Intellia Therapeutics, and Editas. They are working with different companies, and they're targeting different diseases. CRISPR Therapeutics is trying to correct hemoglobinopathies like beta thalassemia and sickle cell disease. Intellia has come up with some really cool things. They have the first patient already this early February this year. They used a CRISPR-Cas9 system to correct. You have to go a little bit into the biology of this particular transerythritin amyloidosis, but they were able to revert back. They actually cured a patient who has this particular disease. Now, I can go a little more detail into this if you're interested at any point or in the chat while the other talks are going on. I want to end before my time ends. So thank you for your attention, and I'm more than happy to take questions. I did run through very quickly, but I didn't realize that the first part took a little longer than what I expected. So, but hopefully this was, gave you an insight a little bit. For those of you who've never heard of CRISPR technology, and for those of you who actually know about it a little bit, gave you a little more interesting pieces of information. Again, thanks for the invitation, and thanks for your patience. I'll be glad to take any questions right now. So I do have a couple of questions in the chat box. My time was until 3 p.m., right?
Yeah, yes, sir. Thank you, sir. We are actually short of time. I think you can
Good.
just take one question. Like, can we insert the P53 gene or any gene of interest that can control or regulate the cancer tumors?
So again, remember, CRISPR-Cas system is actually just able to go in and make changes in an existing piece of DNA. If you want to insert P53, for example, I think there are other technologies. I mean, gene therapy is trying to do that, to actually send in pieces of DNA to be able to express the proteins of your interest. But I think CRISPR-Cas9 is not the system to introduce new genes, but it can actually modify and make changes to existing sequences.
Yeah. Thank you, sir. Thank you so much for a beautiful session and a very beautiful metaphors you have used, like you compared the bacteria to a fort, and a very beautiful explanation of a complex CRISPR-Cas system. Thank you so much for sparing your time and being with us, sir. Thank you. We can move on to the next session. We have our next speaker with us, who's from FABA Academy, who are our partners for the conference. I hand over the session to Purnima ma'am.
Sumati, you can go ahead. No problem.
Sumati, maybe I take over.
Yeah, thank you, sir. Yeah.
Yeah. So firstly, many thanks, Anil sir. I think I wish you were my teacher before I used CRISPR for my PhD. And you really went to the depth, and I really love the way how you presented. Thank you so much. So now we switch gears. No more scientific presentations. No more. Now it's all about career. So we will talk about how you could once you do your MSc, what are the next things? Once you do your PhD, what are the next things? So I'm proud to kind of introduce Nidhi here. Nidhi kind of came up with a beautiful initiative when she was in US, that she thought that we should have some system where every life science students get help. And then she, with the help of Professor Redena, whom you talked in the morning, they initiated something called FABA Academy, so Federation for Asian Biotechnology Associations Academy, to help students like you. So Nidhi, please take over and enlighten us what you are up to with FABA Academy.
Thank you so much, Jagdish, for this brief introduction about what they're going to hear after this very much, science concentrated talks. Both the talks I've been here since, I think Shweta was here. And so it was lovely to hear both of them. And so I would like to share my screen to introduce FABA Academy to all of you. I hope you can all see it.
Yes, Nidhi.
Right. So FABA Academy, everybody. FABA stands for Federation of Asian Biotech Associations. And before I indulge you all into knowing about FABA Academy, which is your guide to a successful career in science, may I tell you a little bit about its parent body, FABA. So FABA was established in 2005, and it is a non-profit, non-government registered organization. It was created to provide a common platform for development of biology across the globe, particularly in the Asian countries. It was designed to act as a facilitator between academia, industry, and government, and promote the cross-border academic and industry collaborations in the field of biotechnology and related fields. Well, since then, since 2004, we have conducted various kinds of events, BioAsia being our flagship event where, the industrialists, the academicians who belong to the field of biotechnology are invited from all over the world. They share one platform to discuss the ideas, to share the upcoming policies and everything. So this is one of a kind of its own event, which is conducted every year in Hyderabad. And then we have conducted events and conferences of various types in various different countries who are our members. We're not only in India, but all other countries also. I will just mention them to you during the end of the talk. So in the year 2020, we indulged ourselves also in a conference that we conducted virtually, which was supply chain challenges of COVID-19 vaccines upon Indian imperative. Many people, including Dr. Renu Swaroop from Department of DBT, Biotechnology India, had joined us, and this was a huge success. So going further, the headquarters of FABA is located in Hyderabad, India, and as on date, there are 19 members countries, including Bangladesh, China, India, Indonesia, Iran, Japan, Malaysia, Nepal, Pakistan, and all others that you can see on the screen right now. And each member country of FABA organizes distinct activities in addition to joining with the headquarters for any common activity. Now, this is the parent body. We felt there was a need to get the skilled manpower to the industries because that is where the gap is. Right now, the prevailing gap is to provide the skilled manpower to the biotechnology industries, to the pharma and biotech industries. What we saw is that the techniques that you are all required at in these industries, our curriculum fails to teach those practical skills. And so to skill all of you in those practical and technical skills so that your resume can be more job relevant, we came up with FABA Academy. We not only focus on skill development, but also on professional development. So the mission is to educate and support students as they explore and further understand themselves and their career options, gain valuable experience, develop as professionals, and launch their post-graduation career plans. Now, as I mentioned, that our curriculum leaves very little space for us to grow professionally. We are definitely taught about the subject skills, the subject knowledge, how things are the way they are, but how to communicate those skills to the industry, how to train yourself in a specific skill set so that you can apply for a relevant job. And so FABA Academy was launched with this idea on 19th Feb 2020, wherein we conducted our first webinar on 20th July, which was a general webinar where the FABA Academy was introduced just the way like I'm doing for you all right now. And since then, we have hosted a total of 26 free webinars, which were focused on career development. These webinars are free and a certificate of participation is awarded to all the candidates. And the basic of these webinars are, they focus on one field of the biotech and pharma industries, and the experts tell you about what is required at your end to be a part of these jobs. So the structure of Pharma Academy is such that we have a skill development wing wherein we focus on training you on the skills which are relevant to pharma and biotech industries. There are hands-on training courses included, some wet lab courses as well as dry lab courses. The second wing of Pharma Academy focuses on professional development, wherein we teach you how to communicate effectively these skills to your potential employer, like CV and resume writing, cover letter writing skills, or mere science communication or presentation skills, interview skills, or even how to network. Now, networking is the key for you to grow in your career professionally. And so you should be able to do it without any problem, without any hesitation. So to know where to start from and how to proceed from there, all these skills are focused as a part of Pharma Academy. Just giving you an idea of what kind of webinars do we indulge ourselves in. So last few webinars out of those 26 that we have conducted so far are, IPR, patents and career opportunities in this field, which was delivered by the expert in this field, Dr. Malti Lakshmi Kumaran. Then we also discussed about in one of the webinars about the emergent COVID-19 and mucormycosis challenges. That was the prevailing problem in society a few months back. And we also had another webinar on present and future opportunities in biopharma and vaccine industries. The recently held webinar was on DNA fingerprinting and its medical legal applications. This is just a snapshot of the kind of webinars we do. Now, as you can see, we do not restrict ourselves only to these fields, but we have held the webinars on agri-biotechnology, even on bio-entrepreneurship, where the bio-entrepreneurs are invited and you indulge with them in an informational interview kind of talk, where they discuss their journey, why they did what they did, and how did they reach to that level. So just a snapshot of what we do on this platform. These are some of the workshops that we've conducted in this year so far. So workshops like basics of chromatography techniques, drug discovery and development workshop, biologics and downstream technologies, biomedical data science and drug discovery. Again, from this is just to give you an idea that we conduct these workshops which are technical skill oriented. Once you be a part of this workshop, you learn about what are industry relevant skills or what are industry relevant topics that you need to indulge yourself in, that you need to learn about and become somebody who is required by the industry, who's wanted by the industry. So we have this ongoing workshop, like at this moment, one of the session is happening. It's on biosafety and biosecurity in microbiological biomedical research, diagnosis, and clinical care. Now, one important factor about this workshop is that it is open to bachelor's level, master's level, PhD, postdocs, faculties, and industry professionals. So it is not restricted to just one category of students here. The whole span, the whole class can join the workshop, and the experts start from the very basic leading to the advanced knowledge. And so anybody can attend these workshops. Once again, some of these could be paid or free for you all, but we also have some membership categories just for your to get rid of any financial burden at your end. Now, this is about the upcoming workshop that we have in the month of November from 26 to 28. It is hands-on training on DNA fingerprinting. So this is going to be a hybrid workshop wherein it would be an online 3-day workshop virtually done on Zoom. But also, if you want, you can avail the physical hands-on training in the center in Hyderabad. It is called Genome Foundation. So this workshop will be happening in collaboration with them. And so once again, it is open to bachelors, masters, PhDs, researchers and teachers from all these domains of life sciences. Well, we are proud to announce the launching of Student Forum. We figured that, when we do things at our end, at the authoritative body end, it becomes a little bit, due to our limited bandwidth, it becomes a little bit difficult to penetrate to all the students or to reach to the student community. And so for that, we launched a student forum. So it is for the students and by the students. The student volunteers come forward, they engage with the experts in the field, they organize webinars and journal clubs for their own community, wherein you can talk, with the experts who has come and has been invited to be the speaker of these webinars or journal clubs. And you can talk to him about the topic or some other relevant aspect also, very much informally. And so student forum activities, it was just launched in the month of June. We've had 2 activities still since then. One was how to build a career in science, my experiences. So this was delivered by Dr. Uday Saxena. He's also one of the executive council member of FABA. He very beautifully presented why did he do, which courses did he take, and why did he take them? After doing bachelor's, why did he do master's? In what subject did he do PhD? And then from PhD, how did he progress into being a CEO today? And then, such experiences, we need to hear such stories, because especially as life science student, such guidance is lacking at some level. And so although we take our courses, we do our bachelor's, master's, PhDs, but however, we feel lost. Even after doing our postdoc, many of us have no idea of where do I want to take my life ahead at, or if that is my career goal, how do I reach to that level? So it is important that we learn from other people's experiences. As a part of student forum activity, we also conduct, journal club. So this time it was on discussion on gene editing through CRISPR-Cas9, wherein Dr. Narasimha was invited, and he very beautifully explained from the very basic about CRISPR-Cas9 system, and people indulged in very informal kind of talk with him, which was very fruitful for the audience who joined. Now, I had mentioned about membership categories. Why do we have them with FABA? Definitely they are paid, but they are paid very much minimally, there are highly discounted workshops if you become the member, and the price is as low as 1,000 rupees for an year for a student. So for this, especially student membership, we just have 1,000 rupees. Once you pay 1,000 rupees, become a member, all the paid workshops start to come at 50% discount for you all. And why do we need this membership? Why do we need some money from you to, first of all, run the organization? Second, it instills a sense of responsibility in the audience. We give all the webinars free, of course. Any kind of webinar that we conduct is free. You're given certificate of participation, as I mentioned, wherein you learn about the basics of careers in different kind of fields, medical devices, agri-biotechnology, IPRs, and varied fields. But there are certain things which need to be charged, so that you are responsible to attend them every day and gain some valuable insights from them. So we have different kind of categories. So for students, we have student membership, which is only annual. And then for individual membership, if you're a faculty, if you're an industry professional, you want to be a member of FABA, you can be a member. Second benefit that comes from being a member is that some exclusive interview opportunities or internship opportunities or any kind of, mentoring sessions with experts in the field are arranged for you. Only if you're a member of FABA Academy. Yes, so if there are any doubts regarding this, please reach out to me. And for more information about FABA or FABA Academy, please visit the website biofaba.org.in. If you have any specific doubts about any of these aspects that I just shared, please reach out to me at nidhifaba@gmail.com. And with this short presentation, I do not expect to take much time of yours. I would like to thank our partners of FABA and sponsors of all the activities that we've conducted so far, because of them and because of people like you who want to avail what we want to give you and gain some value from it, we exist and we continue to strive forward. So thank you so much, everybody. Any questions regarding FABA or FABA Academy, I can entertain. But if you want to reach out to me later, nidhifaba@gmail.com anytime. So
Thanks,
thank you, everybody.
Thank you so much. I'm sure FABA Academy will play a big role in the career and the student's career. So everybody feel free to reach out to Nidhi, and the
Yes.
FABA Academy is also on all the social media channels. Follow them. You'll definitely learn quite a lot.
Yeah.
Hello,
Yes.
Nidhi. Nice to see you.
Hi, Pavni. Yes,
Yeah.
absolutely. After a long time.
Yeah, after a long time. Yes. So I want to request all the teachers, especially I think FABA Academy is more useful for the UG students. We have 30 degree colleges all across the state. So if we have any UG lecturers here, degree college lecturers, please connect your students or make them know about FABA Academy and their activities. Please ask them, as Nidhi was mentioning, they do most of the webinars for free.
Yes.
Their students can always participate and learn. But if there are any specific internships also, they charge very minimal prices when compared to others, because it's mostly for not for profit. So, and we can always have, we always have Nidhi with us who can help us in the other negotiations as well. If we have too many
Absolutely.
students, or if we have an exclusive workshop for a college or something like that, it could be absolutely arranged. So
Yes.
please feel free to
So-
reach them or me, so that I can
Right.
connect you with Nidhi.
And adding to what what Pavni just said is one of the reasons why we charge so minimally. I told you, just to instill a sense of responsibility. But if it is a burden for anybody who is literally serious and wants to be a part of this workshop, see, we are not money making Ponzi scheme. To be honest, okay, we kind of. We have people like Professor Redana in our team, and if he comes to know about anyone who is eager to learn and is financially burdened not to attend because that thing is charged, he waives off the charge. I mean, just straight ahead. So the motive, our motive is to reach the population of students who are non-privileged, who are underprivileged, who do not have access to these skill kind of programs, skill development programs. They are very highly priced elsewhere. And so if you, all the teachers in the audience today, if you want to avail these kind of workshops along with us, how can we reach and know about the workshop in future? Just my next sentence. uh, so please, you can email me nidhifaba@gmail.com. Let me write it in the chat box, okay? Or you can also go onto our website and drop an email at info@biofaba.org.in. Yes, so this is my email ID. Please reach out to me. Just give me an idea of who you are and what do you want from us, and we will get into discussing about it further. Yes. Anything about it, please just reach out to me. nidhifab@gmail.com.
Yeah.
No big deal there.
Thank you. Thank you so much,
Thank
Nidhi,
you.
for explaining us about all about FABA. I think our teachers will reach out to you, or else I think we also have a collaboration which has been long pending.
I know.
We'll definitely, yeah, we'll definitely talk about it and see if we can involve our teachers in few of the workshops which are relevant to the curriculum that they're currently dealing in the colleges. Thank you so much,
Absolutely.
Nidhi. Thank you for your
Thank you.
time, and sorry for the delay in the program.
It's okay, I understand.
Yeah. So without any much delay, now may I invite our next speaker for the session, Dr. Somdatta Karak. Dr. Somdatta, I see you have been given. Yeah. Hello. Hi, Somdatta. Very good evening. So, yeah. So everyone, the participants here, it gives me great pleasure introducing Dr. Somdatta. Dr. Somdatta has a master's degree from TIFR, Tata Institute of Fundamental Research, which is a very prestigious institution in the country. And she has her research experience, doctoral and postdoctoral, from the University of Göttingen in the field of, she worked mainly in the field of neuroscience. And as a researcher turned teacher, let me tell you, I totally understand the excitement that you have in taking science to the public rather than sitting isolated in the lab. So Dr. Somdatta, after her training in science, she started her career as a science communicator, and she works towards making science accessible to all. She creates educational content for students and educators and deconstructs science and technology for policymakers and the public. She currently leads science communication and outreach projects at the CSR CCMB, and that connects active science research and researchers of India with the communities around them. And I also have to thank Somdatta here again on this platform for having two great collaborations with Teas Ferries. First is the Milo CCMB project. So everyone, I think most of our teachers here are a part of the Milo CCMB project. So Dr. Somdatta is the one who played a key role in executing that project or making that project happen.
So thanks again for that. And I also should thank you for another project that we got through your network, which is the Open Field Science Collective. It has been started and the teachers have been participating actively in the workshop and sessions, and they're really enjoying it. Thank you for that. So yeah, we have been collaborating on Milo CCMP project for the past couple of months now. So, and it gives me great pleasure in inviting her again on this platform in this international conference DNA at 150 to interact with us teachers and students and tell them more about, you know, how do we bring science to in the form of stories. How do you tell them to the students from sciences to science stories? That's what she's going to speak about. So without much delay, I hand over the session to you, Somdata.
Thank you so much, Pamini. And I'm actually in a fix now. I mean, after that elaborate and kind introduction you gave, I think you have pretty much covered my talk as well. So anyway, I have a presentation. So let me share the screen here. Yes. So I'm Somdata Karak. I'm a science communicator and I work with CSIR Center for Cellular and Molecular Biology in Hyderabad. And I am struggling to make this full screen. Okay. All right. So as a student, I have been interested in life sciences pretty much ever since I remember, since my school probably. And I studied biology because of that interest. So, from biology, I moved on to neuroscience during my master's and also during my PhD program. And I was doing my PhD in Göttingen University in Germany. And during this program, some other things also happened outside lab. Like I became involved in doing undergraduate courses for the students in the university. I also saw many cool outreach programs that the German universities would conduct. They had something called as the Night of Science, where basically all labs in all universities would, I mean, every university would basically have a night where their labs would stay open for public to just come in and see and talk to the scientists and see what work they would do. And Göttingen is a very rich, scientifically rich place. So there would be things like one could go and see the rover that went to the Mars and stuff like that. And even though I was a scientist, I also found them super cool and exciting. So, and I immediately recognized a lack of a similar platform in India, and that stayed with me. And our graduate program would also take us to different kinds of industries to see what kinds of possibilities and opportunities are opening up. And one of the industries, one of the companies that we went to was an education company. And that was the first ever time I saw that education is becoming, I mean, it was then, I mean, this is like in 2000, I mean, even before 2010. So this is all stories from more than a decade back. And that was the time when education was just beginning to become cool and becoming just, other than that, I mean, for me, education was like, you go to school and then there's a teacher teaches you, you come back and you do your homework. But then I saw here that people were making games for education, people were making different kinds of things that would make learning more interesting. So all of these, I was just picking up here and there. And then it was time for me to finish my PhD. And I had to decide that what I want to do as a longer term thing in my life. And I took some time and I realized that I wanted to work in science education. But then I had never really worked as an educator. So I had to find a place where I could do that. And I started working with Teach for India. Teach for India is a non-profit organization now across 7 or 8 cities in India. And it basically has a fellowship program. It's a 2-year fellowship program. Each fellow has the possibility of working with a low-income community school for 2 years as a full-time teacher. And in those 2 years, you also are encouraged and given the possibility of developing your own area in education. So basically everybody, every fellow kind of thinks what exactly they want to improve in education sector, and they try to work towards it. For me, for example, that was to work in science education. I wanted to understand how science education in India can become better. By better, it means how can it become more interactive, more fun, more inspiring? Because when I was studying science in schools and colleges in India, it was just a textbook studying. And I did not think it was I did not find it exciting, and I did not even find it very useful later on when I studied science during my master's and during my PhD, when I was actually doing hands-on work. And I felt, and I also saw how in other developed countries, for example, in Germany, science education is so much more hands-on, which is completely lacking in India. And I wanted to understand how can we bring a hands-on culture in science education in India. And that was what I largely focused on during my Teach for India fellowship. So what you see here are my students, and I basically during my fellowship, I got a chance to get these students involved in a project in Homi Bhabha Center for Science Education, where they could do real experiments on real questions, real research questions. And that was my first attempt to see how a program like this can even run. And this program was being run by somebody else, right? I mean, I was only facilitating my students becoming a part of this program. Now, what was cool for me is that this program, which is called as CUBE, Collaborative Undergraduate Biology Education, as the name is, it was for undergraduate students, and school students were not really a part of it. And for the first time, I tried making school students a part of it, and that was the most exciting part for me. But then my fellowship ended, and I moved cities, and I basically, during my Teach for India fellowship, I was in Bombay, and then I left Bombay. So I did not stay, I did not continue staying a part of the CUBE program in Bombay. But then I had to take my experience ahead from there to something. And then I had to decide what kind of space would that be. So there were two options in front of me, either the education industry. I told you just that the education industry was growing to make different kinds of learning material, to make exciting education material. So I could either become a part of that, or I could become a part of government space. And becoming a part of government space really excited me. Because science communication and outreach was really blooming in India at the point. So what does science education, science communication even mean? Science communication is basically telling the stories of what happens in research labs to people outside these research labs. And people outside the research labs can be of all different kinds. It can be young students. It can be teachers of those students. It can be parents of these students. And when I'm talking of students and teachers and parents of these students, I'm only talking from the education perspective. But sometimes it is not education. Sometimes it's just information. For example, during COVID, there was so much conversation going on with industry, with press, with policymakers, with politicians. You just name it, and those kinds of communications was happening. And all of this falls under science communication. And as you might understand, that different formats, so each of these stakeholders have different goals. So what a teacher might want to know from a place, from a research lab, would be completely different from what a politician might want to know from a research lab. So these are different goals, and different formats are used for communicating with them. And this is basically what a science communicator's work is in a government space. And now I had to choose what kind of government space I would want to work in. It needed to align with my skills as well as my interest. And my interest was to stay connected with young student population, because that's my favorite stakeholder. So I needed to find a place where they find merit and value in working with young students. So CSIR, Center for Cellular and Molecular Biology, was one such space. We are a research lab in Hyderabad. I hope most of you already know about this. So at CCMB, we interact with a lot of non-scientists all across the year. I mean, in pre-COVID era, about 12,000 non-scientist people would just come to our campus across the year. So that's the kind of space CCMB is. And a large number of these 12,000 people are actually young students. And that's what I enjoy working the most. And I have some kind of training in working with them. So I started working with CCMB. And at CCMB, we have skill development programs for masters onwards people and a lot of outreach programs. And I started taking care of their outreach programs. And so CCMB already has a lot of, even before I joined, I have been working with CCMB now 4 years. So even before that, CCMB had a lot of programs that involved young students from outside CCMB. So when I joined, I started making those platforms bigger and better. One of our flagship programs is called the Young Innovators Program, where students, they apply and they give a test, and they're, top 10% are selected to spend 2 weeks in CCMB, and they discuss with scientists their work, as well as they do hands-on experiments, as well as they just spend a lot of time just thinking about the scientific questions that are addressed in CCMB. So that's one of our flagship programs. And then when I joined CCMB, I started also creating content of newer kinds. So when I was a Teach for India fellow, and because I wanted to work in science education, I thought, I was only thinking how science can become more interactive. But when I became a Teach for India fellow, I realized for the first time how much Indian students also struggle in science because of literacy. So most of the science and interaction around science happens in English, and English is not the mother tongue for most Indians, and it is definitely not a language of convenience for most Indians. So I started making content in science that would be easy to understand. So I started making these things called as zines. The images that you see as zines and comics, which are largely visual. And now I also make videos. So all the kinds of content that I make are largely rely a lot upon visuals. And the hope is that the visuals also help people to understand the situation, the context much better, rather than just heavy, verbose, textual content. Another thing that I have been working at CCMB is talking about some of the urgent problems in society. One of them is climate change, and you see the comics here below, and those are all around climate change. And the other thing is antibiotic resistance. And we have a program called as Superheroes Against Superbugs, where we again work with young children, like many of you here, and we make you aware of what is antibiotic resistance, how we can all get involved in fighting this problem, and also make you aware of how to use antibiotics correctly. So you can also check out our program on the superheroesagainstsuperbugs.com. And we conduct a lot of workshops with school students to make them aware of this program. So currently, a lot of my focus area is on working with teachers to create content for their students. Milo CCMB is one of that example where I have been working. Sorry. So where I work with T-Series teachers to create content for T-Series students. And that is important because every student community has their own needs, has their own strengths, as well as areas of development. And those I get to know as a science communicator only through your teachers. So that's one of the areas that I'm seeing how to make that happen seamlessly and smoothly. Then creating content that is meant for people who generally do not have access to most of the content. So again, that, for me, teachers become the source of information on what kind of content we should be building. And I really hope to, I mean, this third one, I don't think I have really started working on it yet, but that is going to be my next focus area, where I work, where I want to participate in building more neutral and local spaces to discuss science. So what I mean, neutral means, so at the moment, I belong to CCMB and I talk about science at CCMB. So in that sense, I am biased, right? I mean, I'm talking about the work that my peers, my colleagues do. So that's not a neutral space. A neutral space is where I talk about somebody's work where they're not my friends, neither are they my enemies. So that's a neutral space. And local spaces is, at the moment, for a lot of our programs, people need to come to CCMB to know about the work that we do. But that is not that, I mean, probably people in cities can have access to such spaces. I mean, people in Hyderabad city can come to CCMB or other research institutes in the city. People in Bangalore have such other options. Bombay has options. But as you go out of these city spaces of Bombay, the accessibility also decreases. So can we build such local spaces as well to discuss science and questions related to science? So that is something I am currently going to focus working on in the coming year. So yeah, that's it from my end. And you can always stay in touch with me on email. My email ID is here. My social media IDs are also here. And if you want to keep in touch with what CCMB does, we have a new website called as SciTales. So if you go to scitales.ccmb.res.in, you can also stay in touch with all the work that happens at CCMB. And with that, I open up for any questions that you might have.
Thank you so much, Somdata, for sharing your personal journey here with all of us today. I'll tell you the reason why I asked you to join, because we have our school students and teachers here. And as you, I think by this time, you know about our students, these very students and teachers. They're really good at art. So I want them to know that there is a field called science outreach or science communication where they can explore their artistic skills, and using the science narrative, how can they create different art forms. That's what I wanted them to know. And you are like one example that they can immediately relate to or understand how you can take it up as a career. So thank you so much again for accepting that invitation and joining us. So if there are any questions from the teachers, we can take it. So this Dr. Somdata is one more example where you can be trained in science, but you can do something different, which is other than science. So it's not always sitting in a laboratory and doing some sort of experiments. So you have seen different people. Since morning, we have had scientists joining from the industry, where we had Dr. Neha, who is from BioNTech, Germany. So you can do science, you can get trained in science, and then you can join industry, pharma industry, biotech industry. You can become you can work with Pharma Academy like Dr. Nidhi was doing before, or you can be like Dr. Somdata, who is in charge of science communication and outreach. And you also have Dr. Jagdish, who is an entrepreneur. He's also trained in science, but he's not a scientist. He's an entrepreneur now. So Jagdish, so we all have a German connection again here. Jagdish is also from University of Heidelberg. Your university, your attention. Yeah.
Hi, Somdata.
Hi.
Let's catch up sometime again.
Absolutely, we should.
So, sure. Yes. So the main idea is through this conference, we would like students and teachers to know what all different career opportunities you have in life sciences, other than just going for medicine, BDS, or PhD research, postdoctoral research. There are different avenues one can explore. It all depends on your personal interest and what are your strengths that you're good at, right? So, yeah, thanks.
Yeah,
So there are no more questions, I think. So thanks again, Somdata, for joining, and we apologize for the delay.
No worries at all.
Yeah,
thanks for having me here. Bye.
Thank you. Bye. So yes, that's all we have now, but this is not the end. This is not the end. We still have a couple of more activities. The next exciting part is the quiz. I'm sure you must have enjoyed the quiz yesterday. So are you all ready for the quiz for today's session, or do you want to take a 2-minute break and then come back, water break? Or if you're ready, I can start the quiz. Okay, it's 3:42 p.m. exactly now. I'll start the quiz at 3:45. I hope you know the instructions or guidelines for the quiz. You just have to log into menti.com, www.menti.com, and then use the quiz ID that I'm going to display.
Maybe we'll
Enter
give
your
like
name.
5 to 10 minutes break because they have been sitting from a long time, right?
Yeah, that's what we'll start at 3:45.
What time is it now?
3:42. It's 3:42 in India. So
So
I
you'll
thought
give 3
3
minutes
minutes.
break.
Yeah.
Maybe 10 minutes, ma'am. 10 minutes should be good.
Okay, then we'll see you exactly at 5 to 4 or 10.
Okay.
5 to 4.
Maybe we don't close the Zoom. We just turn on the
Yeah.
video and come back. Yeah.
Yes.
Good. Hi, everyone. Are you back?
Jagdish, we said 3:55, so I'm just waiting.
It's okay. But we can start networking in between, right? If somebody has any questions or doubts or anything to share, feel free to put in the chat or open your microphone and speak. I mean, let's keep it interactive.
Okay, Jagdish, I think we can start the quiz. The participants will be slowly joining.
Sure, ma'am.
Yeah, so I'll start sharing my screen now.
Thank you for giving tea break.
People are taking tea break.
Exactly. Much needed break.
Yeah. Yeah. Yeah, yes, yes. So, okay, come on. I hope you all are recharged for the quiz now.
Is the quiz for everyone?
Actually, it's open only for the students.
Okay.
Faculty can participate just for fun, but the prizes are for the students.
Oh, the numbers are going automatically. I think people are tuned now.
Yeah, yeah, yeah. So yesterday we had around 50 participants for the quiz.
Okay.
So today I want more people. I can see 98. So you can remove 8 from the organizing team. So I want all the 90 to participate. Come on, let's have some fun.
So where is Zipti, our Karnool pride?
And I understand teachers like to take tests, but they don't like giving tests. But please try it today.
Shankar Alagunuri link, you have the QR code on the, you see?
Yeah, you have to go to
Screen share.
www.menti.com and then use the code here to enter the quiz. Okay, we have 20 participants. Okay, for those who don't know how to participate or those who have missed attending the yesterday's quiz, you just have to log on to www.menti.com, MENTI, as you see on the screen. Just visit that website and you will be asked to enter the quiz code. And our code for today's quiz is 6107593. You can see it on the screen, or you can directly use the QR code that you see on the screen. Scan it with your camera and you directly enter the quiz. And it asks you to give your name. So please give your name and then you will be in the quiz. I think there's a technical issue on your end because we can see 36 participants who have already joined.
So maybe when they say it's not opening, it's just a screen now. So nothing will happen. You just need to enter the quiz. It says that DNA at 150, and afterwards, once Purnima starts, things will start.
Yes. So the quiz hasn't started yet. We'll start once everyone has joined. So we'll just wait for 1 or 2 minutes for others to join.
Hello, good evening, one and all. I just seen a message from Mr. Ravi Kumar, TGT Bioscience. If you thought you need really some academic guidance to the teachers working in the schools or junior colleges, do not hesitate to write an email to us. Okay, I'm just writing my email ID here, so anyone can write email if you're really interested to carry your say PhD or a higher education. Okay. My email ID is pmamidala@gmail.com. I'm very I will be happy to guide you all. Thank you.
Okay, we have 41 participants. So we have 41 participants, 42. As soon as we reach 50, I think we can start the quiz because we don't want to keep the others waiting. So come on, you have only 7 slots left. And as I told you, you have cash prizes for the winners. Only 6 slots left, guys. Last 6. Yes. 45. So come on, this is bad. I have 1 or 3 participants and only 46 are interested for the quiz. Just take it as a fun activity. Nobody's gonna know your names. You can give any code name that you want. And as you saw yesterday, you're all going to see different animal icons. Zebras, monkeys, smileys, whatever you choose. So please don't have any inhibitions to join the fun. Yes, the number is 50. Now I'm closing and we are starting. Okay. Start countdown. Yes. So we have all the 50 players. Oh my God, we have 54. Yeah, we have 54 now. And we are starting the quiz. All the best, guys, everyone. Be ready. So the first question, as I told you yesterday, again, answering right is not enough. You have to be fast. So the picture that you see here on the screen is Dr. Michael Creeth, and he also has contributed to DNA discovery, and our conference would be incomplete if we don't talk about him. So I want you to know what is that discovery that he made. He is one of the lost heroes of DNA. or the forgotten scientist that we can say. So can anyone tell what is that contribution that he made? Time's up. Oh, I see mixed answers, but 19 of you have guessed it right, I guess. I'm assuming it's a guess. Well, it was a lucky guess. So, as you said, Dr. Michael Creed, he contributed to the understanding of hydrogen bonding in DNA. It was his PhD work that proved that DNA strands are held together by hydrogen bonds. In fact, his model proposed, his DNA model proposed was the closest to the Watson and Crick model that got approved later. And his findings were key for Watson and Crick to come up with their double helical model. So without Dr. Michael Creed and many other discoveries or scientists, we would not have the DNA molecule structure that we are learning to seeing today. So this slide is just an to honor all those forgotten scientists. So today now we know about one more scientist like that, who is Dr. Michael Creed. Okay, time for the next question. I think, yeah. So, but let's see the leaderboard. Who got it right and who got it fast? Okay, it's the spy, K. Sumant. You were the fastest, Sumant. Congratulations. But let's see if you're going to keep your spot with the next question. And here comes the next question. Microbiome is, because in the morning's lecture we have talked so much about microbiome and microbiota. So I want to know how many of you understood what is actually microbiome. We even had a discussion in the participants feedback that we are what we eat, and our microbiome depends on the food that we eat. Okay, I think there's a technical mistake. Microbiome is all the microorganisms that are present in the body, but I think it's showing it as the wrong answer. So we are going to skip this question. Yeah, sorry. I think I should have. And next question. So we have done DNA isolation in the morning. You all have seen DNA. Now I want you to answer, what are the number of cellular barriers to lyse in isolating DNA from animal cells? Today we have seen DNA isolation in plant cells. Mr. Sudarshan has clearly explained what are the barriers and why do you need a detergent to break those barriers. So yes, the right answer for that is there are only two barriers when you are isolating DNA from animal cells, which is the nuclear membrane and the cell membrane. But when it comes to plant cells, you have an additional third barrier, which is the plant cell wall. And you don't find that in the animal cell. So it's relatively difficult to isolate DNA from a plant cell than an animal cell. That's what I want you to understand from this question. Okay, moving on to the next question. Oh, and let's see who is in the lead now. Okay, I'm assuming Deep is Deepti. So Deepti, you're on the lead. Okay. So here comes the fourth question. What is the role of sodium bicarbonate in DNA isolation? We all used it. We all made 1 molar sodium bicarbonate solution. So I assume all of you know the answer, but you have to get it fast. Get it right faster than others to be on the lead. Yes, most of you got it right, which is nice. That means you have attended the session well and you have learned that sodium bicarbonate has a buffering role in the DNA isolation experiment. So let's see who got it fast. Okay. I can see Deepti is again on the lead. Yes, Deepti. So Deepti, Hustler, and Pragya, so far you are the top three. So the next question is your last chance to come to the top or claim retain your spots. Okay. So here comes the next question. And the last question. Yeah. So here you see a comic. DNA is talking to RNA. DNA is asking still single and RNA is saying always. So was RNA being honest with his friend here? So RNA is saying DNA that he's always single. So was RNA being honest? No, RNA was not being honest, because in the morning lectures we have seen from Dr. Shabana's talk and from Dr. Neha's talk that you have double-stranded RNA. We have talked about microRNA, pre-miRNA and all that. So we can see, we have seen that we have understood that RNA, though it's a single-stranded, it can always form temporary double-stranded structures and different secondary structures, as you have seen. So RNA is not always single. So he's lying to his friend DNA. Okay. So let's see the leaderboard now. Oh, I think the Hustler. Okay, the winner for this quiz is the Hustler with 3,248 points. So may I request, now I'm going to stop screen sharing. May I request Hustler to raise your hand? We would like to know who that Hustler is. Yeah. No, if they can raise the hand, we can unmute them and enable the video. Okay, who was the Hustler? Who was that? You can raise your hand because you can't unmute.
I think Sajjanapu Sudharani is trying to speak.
Yeah, okay.
You could speak.
Okay. Okay.
Sir.
Yes, Sudha ma'am.
TSWRS Boinpalli Rajena Sirsilla, madam.
So you were the Hustler?
Madam?
That was your ID on the quiz?
Yes, ma'am.
Okay, congratulations, ma'am. You won this quiz now.
Thank you, sir.
So how does it feel, ma'am? How does it feel to be a part of the quiz and for this whole event?
Very happy to see as first timer, madam. And the session was very, very useful to the teachers to get a complete awareness on this subject.
Thank you. Thank you so much. So what is the best part of the conference that you liked?
Activity, madam. Morning session. Isolation of DNA, I like it very much, but I didn't get the kit. I have to do it practically.
No problem. You'll receive it today or tomorrow. So you can do it with your students in the school. I'm sure they would be excited, and please let us know the feedback once you do it with your kids.
Okay, madam. Thank you.
So yeah, that's it. We are almost at the end of the event. So now, before we start the valedictory ceremony, I request any one of the participants to share their feedback. I can see Swarupa, Dr. Swarupa has raised her hand. Swarupa ma'am, you're on unmute, so you can share your feedback.
Yeah, very good evening, everyone. I'm Dr. Swarupa from TSW RDC. Yeah, am I audible, ma'am?
Could you please be a little louder?
Yeah, sure. Yeah, very good evening, everyone. I'm Dr. Swarupa from Social Welfare Residential Degree and PG College. I'm very happy to participate in this conference, which is very well organized, and all the sessions were very interesting. Basically, you have the interesting thing is you have come up with very unique paths for hands-on experience in isolation and extraction of DNA. And even Mr. Sudarshan have explained principle behind extraction very well. I hope now students know the principle and also the procedure. So because they have done live by seeing the video and taking Sudarshan guidelines. And also even our PG students have also participated in poster presentations. Now they had an experience how scientific research can be presented. So I hope this session overall conference, DNA at 150, is definitely useful for the students and also to the teachers. I am definitely looking forward to more sessions like this. Thank you for organizing such a wonderful conference. Thank you overall T-Series team and especially Pavani. Thank you so much.
Thank you, ma'am, and thank you for your valuable feedback. Any other participant who would like to share that feedback quickly before we go to the valedictory session, where we have prize distribution and announcement of winners?
Chekuri Padmavati has raised hand.
Okay. Padmavati ma'am, you can go ahead and speak now.
Yeah, good evening to the organizers, Dr. Jagdish and Dr. Pavni. In fact, it was an extremely innovative way of how you conducted it, including all the school students, their teachers, school teachers, degree college lecturers, both, mostly from the Telangana state. Actually, I work in Vagdevi Degree and PG College at Hanamkonda. In fact, it was extremely exciting, and though I had a brief idea when I saw the program sheet, but experiencing it, I felt it was extremely innovative. I think this is the first time I have been seeing a particular seminar or something being conducted in this way. And just imagine, we never imagined that we could get a kit and do it at home also. Of course, I usually ensure that my PG students do the DNA isolation. It's a regular practice, but still getting it at home, I'm sure the students, especially the school children, would have been extremely excited. Whether it was the figures that you had organized, or whether it was this quiz or anything. And even the lectures, they were so crisp and so easy for assimilation, directly prepared, like it was more suitable to the students at large. So I really congratulate all of you, Dr. Reddanna, Dr. Praveen Mamadala too, who have been your support, and very nice to have seen all this. Congratulations to each and every one of you.
Thank you so much, ma'am. Thank you for your valuable feedback. It means a lot to us, and with your encouragement, we look forward to conducting more activities like these in the future. And last participant feedback we are going to take from Ravi Kumar, TGT Bioscience. Stop.
Good evening, ma'am.
Good evening, sir. Please go ahead.
Good evening, everyone.
Yeah.
So this is really an amazing session. I have never saw such a session throughout my education. Really, it is a good session, ma'am. We have got a chance to update ourselves, though we are masters in biochemistry and biotechnology and other fields like other teachers, but we have been cut off from this research, all those things since many years. Now we have got a chance to update ourselves, and not only that, even students also got a career guidance through this session. And most of the students think that if they don't get a seat in MBBS or like a medicine, it's a dead end, they think that. But now in this session, students are aware of different career paths, and they have a wide range of choices in the field of science, and they can choose science as a subject, and they can build their career in this. So it's a good session, ma'am. We hope like these sessions would help us in future to the students and teachers also. So thank you, ma'am. Thank you very much.
Thank you so much, Ravi Kumar, sir. We are glad that you enjoyed these sessions. So thank you everyone. We can't take due to lack of time, we can't take all of your feedback. But we are assuming that you all had an enjoyable session and great learning over the past 2 days. So without any delay, I would like to start the valedictory session now. So as they say, all good things come to an end, right? So though it is really exciting, it's really enjoying, but we have to conclude this session too. But again, we have to remember that all endings are beginnings for new things, right? It's not actually an ending. So though we are ending here, we are going to begin or see you again with a great event like this, or better event if possible. So let us quickly start the valedictory ceremony now. So before we start, I request our Joint Secretary, Dr. Praveen Mamidala, who is also the convener for DNA 150, to give his valedictory address.
Thank you very much, Dr. Pavani, and I'm really so excited to talk to, particularly with the students. I can see the Social Welfare Residential School Mother. Hello, the Mother girls, so excited, and I'm really happy to join today's valediction meeting. I don't know how time passed just like that. It's already 2 days we have passed out, and I hope everyone has enjoyed this 2 days international workshop, DNA at the rate, celebrating 150 years of DNA discovery. Special thanks goes to Jagdish, Jagdish, CEO, young CEO. I'm sure in future he should, or I wish that he should become a billionaire or trillionaire. It's all innovative ideas. And also to Dr. Pavani, who is like my left hand and right hand in both of academics and administration. For the meticulous planning in a way in designing of this conference come workshop. In my experience, honestly saying from bottom of my heart, I never seen such virtual conference combined with the of a workshop, hands-on experience. Just now, Chinkuri Padmavati Garu from Vagdevi College rightly said, yes, it is really a new idea. Thanks to Cambridge also here, who has posted at least today. If not, it's gone. So finally, it reached destination today, and everyone has enjoyed the hands-on experience of isolating the DNA. And before I say something, I should not forget, and I should say special thanks to Secretary Dr. Sri Ronald Rose, IAS officer, who is always with us, and he felt so happy with the yesterday feedback. He already got feedback and said, Praveen, wow, school children have joined this program. I'm so excited. Why I'm saying or why I'm focusing on school children is, unfortunately, is until completion of masters in science, no one understands what science is. That is unfortunate. When we come back, what is science? As a science teacher, what I feel is science is a domain where every student must understand the fundamentals, followed by a very close observation, and followed by a meticulous experiment. See, you should understand the topic. For example, just now Pavani has shared the quiz questions, very interesting questions. Who is this guy, or what is buffer? What is like microbiome and RNA world? Though they complete the MSc, people do not know what are the different small RNAs. What is siRNA? What is microRNA? What is the exact role of, no one talks about tRNA or rRNA, something like that. This RNA world itself is a very big world. It's very small, but it's very big, really. The topic is also an awesome topic. What I want to say is, when you don't have a clear understanding in science, then you do not observe. Observation is very important in the science, because that observation made Friedrich Miescher back in 1869 to discover DNA. An experiment, repeated experiments until you come out with some solution, and that solution must resolve the societal issues. This is very important, my dear friends. As a science teacher, what I can tell all of my fellow colleagues or students is, budding students, we have school students, we should plan our experiment in such a way that will definitely resolve several hundreds of issues in the society. At the same time, you should make into business, just like Jagdish, how he's trying since last few years. Very few people, particularly biologists, they go into business side. Very few people see why DNA is still Greek to us is only thing is biologists will always try to remain as biologists. That is wrong. Biologist should be a chemist. Biologist must be a physicist. Biologist must be a very good mathematician. Biologist must be an all-rounder. Understanding science, linking to society, then only you can see some very good innovations, my dear friends. So I hope the networking, the connections which has made through this conference, and I'm sure that our students has taken it very much, and definitely in future uh, teachers and students will be connected to our higher education wing, and definitely if at all if you have any doubts with regard to the biology, particularly life sciences. When I say biology, life sciences, career-wise or future, whatever the programs you want to do, please do not hesitate. Just send some messages in the chat box. So please try to get back to us, and we are here to resolve all of, or at least to suggest you or recommend you what are the future courses you have to take. So if you see the participants list, it is almost close to 100, 103. In any given conference, you never see this much of strength at the time of valediction. People will see, they will leave the conference just like that. But here you see the number is same. I observed, no, in the morning session also it was like 143 when the lecture started in the morning, and up to the lunch time the number is same, 143. No one is left. That shows the powerful speakers from Australia, from India, and also from Germany, UK. Jagdish is managing the show, because for us 10 o'clock is okay, but for him 10 o'clock is very early morning, and but he came by the time. Thanks Jagdish, thanks a lot. So everyone has really made a fantastic work for the 2 days. I really appreciate the organizers. When I say organizers, it's not me and Pavani, but we have some academic fellows like Shiva Roy, and we have some other officers like Resina, and also some other organizers from the teachers from my side. We have several teachers working on this DNA Drill 150. Tons of thanks to all the people behind this project, and also the complete higher education wing making this as a grand success. I'm really happy. So as I told you that if Jagdish is with us for next 1 year, so I want to do version 2, 3, 4, 5, so that at home, if Cambryonics help us, I have because Jagdish, I want to tell you one thing, and also to all the teachers. For example, there are some teachers from private colleges and degree colleges also. Till date, up to MSc level also, people do not do any cloning experiments, unfortunately. They will only write in textbook, in the practical record book. Things I think they may do, but I'm sure that in the degree colleges and PG colleges, even in the state universities also, they don't do gene cloning experiments. If you can plan such experiments in the next short, how to do cloning, or how to some understanding the like computational analysis of DNA, very simple things. If you can do in the future, I think students get a lots of benefit. Thank you very much, my dear friends. Thank you one and all. Bye.
Thank you so much, sir.
Yeah. Thank you so much, sir. Thank you for the address. Now we have a really interesting session. So that is prize distribution for the participants. Now, may I request Jagdish to announce the winners for the poster and SciArt competition? Over to you, Jagdish.
Oh, sure. It's a bit weird for me to give prizes, because I always organize events and hand it over to others. But for the first time, I need to announce it. So For the people who ever have presented posters and art.
Okay. Someone is on mute.
ఆయనకేం తెలియదు.
నిజంగా నాకు తెలియదు. Well, anyway, so we did a lot of discussion, like whom to choose, who are the best prize winners, whether it's art or the poster. Then we came to a conclusion that we are not doing this kind of competition for others. We are doing everything to encourage you guys. Like, we want you to learn how to do poster presentation. We want you to think about what is science and how to make scientific art. So as some teachers told in the in their feedback, this is the first kind of conference that people are doing more interactive sessions, like isolating DNA or DNA art or poster presentation, quiz and everything. So the main logic of the conference was this: we celebrate DNA discovery 150 years. We will add a little bit of talks, but we will do more of the events. So that's why we decided we are not going to give prizes for first, second places. We are going to give prizes for everybody. Whoever has presented your posters, whoever made the scientific art. We really don't want to be judgmental here. We can't judge your especially with the online environment. We have no clue what really worked well or not, because sometimes, especially in the first poster we have seen yesterday, there were technical glitches, and we don't want that to be your decision maker, to choose you the best or not. So that's why we decided that whoever have participated, you are the winners. So participation is better than the prize. So there is this proverb, right? So we decided everybody will get a prize, and we will think what will be the prize. Everything will be sent to your home. Maybe it's a good book about DNA discovery, or some kind of kit from Cambrionics or any other company. So guys, like, whoever has presented, a big claps to all of you, because you dared to really present, and most of them didn't present. So congratulations to everyone. Yes,
Thank you, Jagdish. Thank you. So please, so the whole idea, as Jagdish said, was to encourage students to participate. So we are going to send a certificate of appreciation for everyone, those who participated in the posters and art competition. And we are going to send a book prize or a memento or a kit, whatever we find more suitable and useful for you tomorrow to pursue science as a career, or which can inspire you to pursue science. We are going to surely send it to you. It will be shipped, or else you all have all our email IDs. You can write to us anytime. Okay, so we are not leaving without that. So that's what we decided, actually. Instead of prizes, initially we thought of giving prizes, but then we wanted everyone to be happy and to be participating. And you keep participating in every conference, present a poster everywhere. That's how you learn presenting posters, and that's how you're going to enjoy and learn more about the research that's happening. So now, but we have winners for the quiz competition. We are not going to give away prizes to everyone who participated. So now may I request our Joint Secretary, sir, to announce the quiz competition winners.
Yes. Okay, but I think they might be knowing about the winners, right? Because already we have in the list, but formally. Okay. So the quiz on the day one, the first round goes to Supriya Maripeddi, MSc Biotechnology student from Kakatiya University. Congratulations, and I am the alumni of KU, my dear friends. So, Supriya, congratulations, beta. Okay. And on the second round, on day one, it is medico doctor. Wow, I'm so happy. Students generally, medical students do not join biotechnology programs, but I'm sure that in future is genomics, medical genomics. So congratulations, Dr. Deepti. And yeah, on the day two, the quiz program is Sudharani ma'am, from which school she is. Okay. Mrs. Sudharani, congratulations, Amma. Well, good job. I know, being a teacher in the social welfare school, that too, you might be a TGT or TGT. You need to spend lots of time. Still, you have made it. Yeah, congratulations, Mrs. Sudharani. Congratulations to one and all. Yeah. So, what you're going to give them?
Cash prize.
Cash prize, how much? Yes. So, friends, you are going to get 1,000 rupees through Google Pay right now.
Okay, you can drop it in the chat box.
So, please share your mobile numbers and all these 3 winners will get 1,000 rupees cash, not cash prize, Google Pay.
Yeah. So I'm
Yes.
ready.
Our co-convener is ready. It's from your side. Please share your mobile numbers and you are going to get money. Before vote of thanks, is it possible?
If they send the money, it's possible.
Okay, so they will take some time because next is the vote of thanks, I guess. Over to Dr. Pavani.
Yeah.
As Praveen sir said, please drop in your mobile numbers to Shiba Roy, who is in the chat, and we can give you the cash prizes right away. Okay, so I think Praveen sir has already mentioned everyone who are involved in this conference and who are involved in making this event a great success. But again, let me formally thank everyone who is a part of this event. Good evening, everyone, to one and all who are present here. And those who could not join as well, especially our Honorable Secretary, Sir, Sri D. Ronald Rose Garu IAS, and all the participants. It's such an honor for me to get the opportunity to thank you all dignitaries. On behalf of the organizers, T-Sweries, I extend a warm thanks to the people in the gathering. I would like to express my gratitude to all the esteemed delegates of the webinar for their presence and contribution to make this webinar a grand success. I extend my gratitude to our Honorable Secretary, T-Sweries, Sri D. Ronaldo Sir, for leading us and this organization towards success. And I have no words to thank our Joint Secretary for Higher Education, Dr. Praveen Mavidala, who is also the convener for this international conference, for his continuous support and guidance. His energy and the enthusiasm at the workplace is infectious, and it keeps motivating us to do better every day. Thank you so much, sir. And a special thanks to Dr. Jagdish Gandla for providing his valuable inputs to make the webinar take this shape and making it truly international by bringing in speakers from the US and Europe. I extend my gratitude to Professor P. Redana, Executive President of FABA Academy, for his guidance, and we look forward to organizing more collaborative events with FABA Academy in the future. And I must definitely thank my organizing team, my colleagues, Dr. Sumati and Dr. Purnima, for their support, and our higher education wing, which is like a family at the workplace. And big shout out to our Super Shiva Roy. Thank you so much, guys, for working hard for the past few days to make this event very successful. I congratulate all the winners as well as the participants for their active participation. And thank you so much, everyone, once again, for making it a grand success. On behalf of organizing committee and as a co-convener for DNA at 150, I now conclude this session. And thank you everyone for participating. And we look forward to meeting you again in another exciting and interesting session. So Jagdish, do you want to say bye?
Yeah, yes, ma'am. Yeah. So maybe I should also conclude. So very big thanks to Pavani ma'am, Praveen sir, and all your team like Shiba, Purnima ma'am, and Sumati. Very well organized everything, and I somehow feel so satisfied running this conference. I mean, my events are usually big events like with money and everything, but this gives me immense satisfaction than doing those events. So actually, I was supposed to have a networking session today, but I kind of skipped it because we were running late of time. What I want to talk was like life science has good career, so there are many opportunities, and especially after this COVID pandemic, life science has rejuvenated. So there's a lot of companies that are coming up, a lot of initiatives that are coming up. Only thing that you have to do is that go out and get to know these things. So you go out in the sense like you don't need to go into COVID areas. You go out in the social media platforms like LinkedIn or Twitter, somewhere you will get to know things and start speaking. Like you kind of request people like, sir, I would like to know, madam, I would like to know. So when curiosity is your main weapon, I think there is a lot of career in life sciences. So I even saw in the chart, like one madam wrote like now she was confused whether her daughter should have in mathematics or life science as career. Now, after coming this webinar, now she decided that the life science has career. I mean, I truly believe life science, especially with the biotechnology, we didn't even touch the tip of the iceberg. There's a lot that we have to do, especially India has a huge role to play. I mean, in Europe, what I see is that we are talking quite a lot about digital health, artificial intelligence, machine learning. And of course, next 10 years, 20 years, we will see a lot of trends in digital health. And if you want to make an immediate career in life sciences, I would recommend you to do this kind of courses, like as Venu said yesterday, learn coding. Even though you are a life scientist, you did by PC and everything, but you can learn coding very easily. I mean, I don't code, but I could make career in life sciences. I did my PhD, and now I worked for European Union, and I'm kind of now helping other companies as well. So if you want to make a good career immediately, coding is like learn coding and learn some programming and everything. You have very good options. But if you want a long-term career where you can impact many lives, then life sciences, biotechnology is what is a crisp. So there's going to be many revolutions that we will see. As Anil Sharma said today, CRISPR, the babies that the Chinese guys kind of edited, it's not legal, but I'm sure it will be legal soon, where we'll be editing our babies, like if there is any genetic mutation. So we could use CRISPR technology or many other technologies that will come in future. So with all these things, what I want to conclude is that be open in the sense, like be ready to mingle with people and get to know what's happening all across the world. And never underestimate yourself, even though you come from Telangana or Andhra Pradesh, or even from rural village, belonging to backward caste or scheduled caste. It really doesn't matter. As long as you are curious and want to contribute stuff, I think whole world is open for you. They are waiting for you to come and innovate, because there are many things that has to be addressed. Look, like tuberculosis, we don't have a vaccine. Malaria, we don't have a vaccine. Dengue, we don't have a vaccine. We have more people dying from malaria than COVID, actually, But the thing is, it never got our attention, and we never cared. That we think that America will come and invent a vaccine for us. But why would they do? Because the malaria is not there in America. Malaria is not in Europe. Only we have it in Africa and India, and we have to think about it. So we have to invent, we have to redesign what we have. So I think it's our responsibility as biologists. We are getting trained in biology. It's not the government's responsibility. We have to do this stuff. So like this question that you need to ask yourself, what can I do for my country, for myself? And if life science is your tool, I think that is your best weapon. So go and hit the milestone. And with this, I would like to conclude, and special thanks to Pavani ma'am for bringing onto the stage. I really enjoyed it.
Thank you. It's my pleasure. And I'm glad that we collaborated and we could pull this off. And thank all the participants for making it a huge success. It would not be the same without. Thank you so much, and see you again in another session of learning. Definitely, we promise we'll try to bring one more interesting session for you all. So keep learning, keep enjoying science, and keep teaching science, and make kids discover the joy of learning and joy of science, just like we discovered through science. Yeah. Thank you everyone. Have a good evening.
Everyone.
Yeah.
Ma'am, since we are stopping, if somebody wants to leave, feel free, or somebody wants to talk, maybe we can open the mics, no?
They didn't send their mobile numbers, huh?
See, I mean, the conference is concluded, so feel free to leave. But if you want to speak, maybe I'll open the mics, so feel free to talk. So who wants to speak? Mamta Garu, I'm trying to unmute, but it's not working. Yes.
Okay. Good evening to respected Praveen Mamidala sir, Pavani ma'am, and Jagadish sir. So I'm so happy. Huge thanks to the Praveen Mamidala sir to providing such a nice informative session. By that, to reboost all the teachers' minds. And anyways, either it may be teacher or the students. So we are the learners in our life. So whatever things we are every day going to be learned, it's a drop of the ocean. So thank you so much to the sir by joining with the CCMB and providing interaction with such a great scientist and many doctors yesterday and today also. I'm so much glad for that. Thank you so much to the Pavani ma'am for patiently handling and organizing all the session, as well as the madam also responding to the messages in the group, CCMB schools group, WhatsApp group. Then thank you so much to the Jagdish sir, organizing such nice program. So here, thank you so much to everyone, those who are always trying to reboost the teachers, to encourage the teachers. So I want to give my personal honorable thanks to our Praveen Mamidara sir. Why? Because sir provided on-duty facility to all the teachers. Why? Because from morning to evening session, it's very difficult to be in the school, and continually we can't able to enjoy and make a note of all the information every day, which we are going to share all such information on Monday when we are going to school to share all such things. Thank you so much, sir. And thank you so much to the Sudarshan sir for the process of DNA isolation. So it's very, very huge and very interesting, and tremendously we are very happy, even including students also. So I noted on all the process, everything, but unfortunately I did not got my isolation kit. I hope by tomorrow or today I'm going to get it. While that I will show you to my students. Thank you so much, sir.